me a note about that, about so there's like 130 registrants, and the number of people in the Zoom room for each session has been a lot less than that. But I liken it to, I mean, any given conference, you add up the number of people in the hallways and the number of people in the session rooms. I think it probably works out to about the same.
Well,
And
we do have the YouTube streaming as well.
right, and I need to check the stats on that.
Have you given Christoph the right to share his screen?
I will do that right now. Yep.
All right.
So you ready to go?
Okay, so just for everybody that's here, this is Christoph Müller-Dickmann, and he is one of the senior people in the ESRF Structural Biology Department. And he's going to tell us about how the ESRF can serve us and our community.
Are you starting right on time?
Aren't we starting right on time?
You're making me dizzy with that.
You mean it's hard to start on time?
We can
No,
wait
that's fine.
a couple of minutes to see when more people will join.
Right, no problem. Do you want me to unshare?
No. You can go ahead and share because there's the live stream. So
All right.
actually, I don't know. We could start right on time. That's fine.
All right. Good. So, good morning to everybody. What I try to do in the next half an hour or so is to show you a little bit about the facilities of the structural biology at the European Synchrotron, and actually how you could profit from the facilities, well, how you and your research can profit from the facilities, what we are offering. So, but before starting, I would like to acknowledge all the people which is basically behind the work which I'm presenting you. So you see, there's quite a lot of people from the ESRF involved, but also from other institutes which are on the campus, like the EMBL, the IBS, and the ILL, and an organization which is called the Partnership for Structural Biology, PSB, which brings along even the University of Grenoble. And talking about these 4 institutes, they're all located on a campus which is called the European Photon and Neutron Science Campus, or EPN. Well, and you see the most prominent one is the eye, which is the ESRF. This ring-like structure, you have the reactor, the neutron reactor, the ILL. And you have the EMBL outstation Grenoble, pretty close to in between the ILL actually and the ESRF. And the French Institute of Structural Biology, which is on the campus as well. So there's quite a concentration of structural biology and structural biologists on this campus. Roughly 600 scientists are working in structural biology here. All right, but focus on the ESRF. The ESRF is actually, well, it's the European Synchrotron, but you can see that there's actually member states which are not Europe. Yeah, prominent ones is South Africa, India, and Russia, but also Israel. 22 partner nations it has, of which one is South Africa. You see this one as an associate country. Quickly going through the ESRF founded 1988, by 11 member states. As you've seen in the previous slides, this has increased, almost doubled with time. First electron beam 4 years later, and 2 years later, then in 1994, it was inaugurated with 15 beamlines running. This has almost tripled within 4 years, and then was running for roughly 10 years with these 40 beamlines. And from 2009 onwards, there was quite an ambitious upgrade program going on, which was making, the ESRF was the first 3rd generation synchrotron, and it was making the ESRF a 4th generation synchrotron. I come to this one in this slide. You see that there were 2 phases for the upgrades. One phase, which has finished in 2015, was mainly focusing on the upgrade of new beamlines and the construction of experimental hutches, which you see in magenta there, and the upgrade of the ESRF machine was starting in 2015 and has finished just recently, which is a complete reconstruction of the storage ring. This ESRF is called ESRF Extremely Brilliant Source, so ESRF EBS. Well, and what happened in this redesign, is actually that the complete storage ring was taken out and replaced by new cells, which are much more complex than it was before. And you see this one in the picture below, the old setup compared to the new setup, which is called hybrid 7-band lattice, which is actually composed of more magnets and more vacuum chambers. All this one done actually to make the beam, which was before in the old ESRF, you see this one on the picture on the bottom left, rather horizontally broad, to bring this one back to a very low divergent and very focused beam, as you can see on the right one. With this one, you see on the right the evolution of the brilliance versus the years. You see that the ESRF has increased its brilliance quite considerably with this upgrade. So now the question is, of course, what is this ESRF Extremely Brilliant Source good for structural biology? This I try to answer in the next couple of slides. Bringing up something which is rather old from, well, that is cited from a paper which was published in 2011, stating that crystal structures, of course, are just a static snapshot of the protein. You may gain some additional information having a closer look at relative B factors and alternate conformations, but especially if you go for cryogenic temperatures, these alternate conformations may be frozen and not really be visible. So room temperature certainly has an advantage because it gives you some additional information over the cryogenic temperature structures that you have. And one of the prominent examples is in this Fraser paper from 2011, which I have mentioned, where you have a glutamine residue, which actually the active conformation could only be seen at room temperature and not at cryogenic temperatures. So, but we all know that working with ionizing radiation is actually, and room temperature is actually not the best combination. So radiation damage is much larger at room temperature. So that means that you're almost obliged to do serial crystallography to routinely obtain structures at room temperature. On this slide, there's maybe a little bit too much, but what I want to show you there is actually what can you do at a synchrotron site? I mean, you certainly cannot go to see bond breaking and side chain rotation because the time line that these things happen are far too fast for what we can observe here. But you can go for this regime. You can see domain movements, allosteric transitions, maybe the hydrophobic collapse and slower loop or hinge rotations. What you can also do is these pump probe experiments that you trigger a reaction at a certain time, and then with a delta T, you probe or you take an image of your sample and you try to get snapshots over the complete course of the reaction cycle, for example. And this is actually done in an experiment still with the old EBS, where some of our scientists were interested in seeing how a light state of a photo sensitive protein is built up. And what they did is they exposed the crystal with a certain light to trigger this conformational change. And then they took from different crystals, very low dose complete data sets. And they did this for a total of 88 crystals. After that one, they merged just the first 15 image, and the first 15 image corresponded to something like 63 milliseconds. And if you go through this one all the way through, you have basically built up a picture of your reaction cycles in steps of 60 or a little bit more than 60 milliseconds time resolution. So all these experiments, oh wow, this one is just to show that since the different crystals need to be merged, you need to make sure that the isomorphism between the different crystals is sufficiently high, or sufficient, well, the isomorphism is good enough to merge them to get a good picture. And this is what you do with this hierarchical cluster analysis picture, which I put in there. So these kind of experiments, which I have mentioned, room temperature, serial crystallography, and time resolved experiments, they are basically we try to address them by currently constructing a new beamline, which is the old ID29 beamline, which you can see in this picture down here. These are the two old hutches which were existing. This was the experimental hutch. Now it's an optical hutch, and you go pretty much down outside of the ring. We are here currently around 110 meters from the source, where you have a rather small beam. And if you look there, well, there's a sample preparation lab, plant, a data analysis room, and a control cabin. And if you look at the specification of this beamline, you see the most important one is that this is a rather high flux beamline intended to deliver 10 to the 16 photons per second in a beam which is less or equal to 1 micrometer. So equipped with a new detector, which we are currently commissioning together with the PSI, which is a rather high speed, 1 kilohertz readout time with 1 microsecond integration time. This should bring us in a situation where we have a time resolution going down to 1 microsecond in uniform mode. Well, and along with the commissioning of such a beamline goes, of course, the commissioning of new sample delivery systems like fixed target systems, which are already existing, this is a high viscosity extruder where the crystals are dissolved, or not dissolved, but inside a high viscosity carrier material, such that the flow rate is reduced and you have the chance to hit more crystals on the way they are coming out of this little tiny nozzle. You see, this is the X-ray path. Well, sorry, that was one too fast. So all these new delivery systems, they need to be developed or are being developed currently for this new beamline, which is dedicated for synchrotron serial crystallography. Well, but ID29 is not the only one. You may know that we have a couple of other beamlines, which I will quickly introduce to you. They are in this picture depicted as or written in black, while the ancillary facilities are in blue. And you see that there's quite a lot of ancillary facilities around. And I think that these ones are actually quite interesting and for you, because they can support your research on the structures quite considerably. So coming back to, we have 8 end stations running, 6 for diffraction or macromolecular crystallography beamlines, 1 solution scattering BioSAX beamline, and 1 cryo-EM facility, which is located around here and in this location, but you will have a picture later on. It's a very rough overview over the X-ray diffraction beamlines. You see that we have a microfocus beamline, for example, on ID23-2. We have a completely automated beamline. I will talk about this one a little bit later on Massif 1, which is a mail-in facility. We have a mini beamline, roughly 15 microns in diameter, and 2 energy tunable beamlines between 6 and 20 keV, all equipped with fast readout detectors, as you can see on the detector samples. We have all the beamlines, the diffraction beamlines equipped, or most of the beamlines with apertures such that we can tailor the beam size to the crystal size. All are equipped with a FLEX high density pure sample changing robot, which accepts either Unipucks or these ST3 type vial pucks, with 2 exceptions actually, that ID23-2 and Massif 1 for geometry reasons only accept the Unipuck system. All the other beamlines accept whatever you come with. So on all the beamlines we have different grippers. This is the gripper, for example, for the vial type things. For the Unipuck, we have either single or double gripper. We have 14-second exchange between these between samples. All right, I come to something. Our control software is MX-Cube, and MX-Cube is actually a big collaboration within Europe, and many of the synchrotrons in Europe actually have this MX-Cube installed, such that for you as a user, it's easy to change in between these synchrotrons, because it's you're getting used to this software. So you see that you have a very nice overview over your sample. This is a sample mounted. You have the video brick. You can control the sample changer very easily. You see which of your samples have been mounted. You can do X-ray centering to center your crystal, especially if you have small crystals, based on X-ray diffraction with very low dose. So all the normal thing that you're used to. And everything is then displayed in something which is called the ICE-BY-B database. And we have a GUI which is shown, which is called EXI, which is in extended, is also a big collaboration between different synchrotron sites, mainly in Europe. And you have an overview over your experiment, which gives you right away a good indication whether your experiment is going into the right direction. For example, here you have a characterization done. Sorry, you see in this picture that composed of 4 images, 90 degrees apart. If you click on these images, you see that they were successful. You get an overview. You get all the information that's associated with this characterization done, starting from down here. I think it's I can't read it. That's the 4 images which have been taken. You see the snapshots of the images, and you see the data, or the data collection strategy, which is calculated based on these 4 images. If you go further down, you see that you have a nice tool to check that your data collection goes or went correctly by checking, for example, that your visible resolution, or the number of spots collected per image, is not declining over time. So a nice way to see that your data may not be polluted by radiation damage. All the data collections done are automatically processed using different pipelines. You get the result of each of the pipelines. You have the possibility to download the results from these ones, they are classified as the best one based on different criteria like the R merging statistics and the A over sigma. You also get the possibility to do molecular replacement based on the nearest cell. You see that if you, up here you have a little icon which says molecular replacement. There was some success, and if you go, that's stated in IceBaby or in XSEA as well, right underneath your data collection brick, and you have the possibility to click on one of those little images here, and you get a very rough view on what the molecular replacement solution looks like. So I already mentioned in the beginning that we have automatic workflows actually on all the beamlines. Massive One is the beamline which is exclusively using these mail-in crystallography. And if I concentrate on this MXpressE workflow, you basically see that this is a chain of different things working together. So you mount your sample, you optically send to the loop, you search for with a low dose X-ray centering for the point of highest diffraction, collect 4 images, calculate your strategy, and according to the plan given by BEST, and you collect your diffraction data. And mount the sample afterwards, mount the next sample. And with this one, you can go through all of your samples that you're interested in. That's especially interesting for those projects where you have, for example, look for ligand screening or fragment screening or something like that, but not exclusively. So it's very much developed on Massive One, but it's available on all of our MX beamlines. Some words about the sample environment. There's a lot of things that we can mount in our sample environment. All of our beamlines are equipped with a mini Kappa goniometer head, so you can change the orientation of your crystal. Also for doing, for example, Z experiments, if you're interested in getting Friedel pairs collected close in dose and time. We have on some of the beamlines the possibility to mount crystallization plates or a microspectrophotometer. I'll come to this one in a second. Dehydration devices or to keep the relative humidity at room temperature controlled. We have devices such that which we can put on all the beamlines as well. We have on some beamlines the possibility to mount these high viscosity extruders, for example, or microfluidic chips, or crystallization chips as shown down in this picture here, where we can mount the small crystallization chips directly on the beamline. I mentioned that we have one solution scattering beamlines at temperatures between 4 and 60 degrees, mainly at room temperature, equipped with online chromatography, in which you can check for your complex, how this looks like. It's equipped with a very versatile sample exposure unit, also giving you the possibility to use, for example, spectrophotometry to check for UV or UV-Vis absorption. This also can run with microfluidic chips in this sample exposure unit for different types of experiments. And if you're interested in this one, please just contact one of those working on BM29 to get more information. We have the possibility to couple the solution scattering with neutron experiments or scattering experiments using neutrons. So SUNS experiments during one drip that's done in collaboration with the ILL, which also have an automatic sample changer and online chromatography. These ones you use usually in this contrast variation is a quite nice technique to get more information about a complex which you have. And usually this is done that you have several days of SUNS beamline at the ILL and then come to the ESRF to perform a SAX experiment right after. I also mentioned that we have a cryo EM facility, which is equipped with a Titan Krios G3 microscope. Currently still equipped with a K2 detector, but this is being replaced in a month's time by the latest K3 detector. Access you get either through back access, but for you guys, most likely it is rolling access proposal, very easily done and described on our web pages. Each experiment is evaluated by our beam time allocation panel and then granted beam time. So usually to get access to the microscope, you need to come with an already characterized sample to justify the use of the Titan Krios. But for those which do not have a very good access to screening microscopes, we have put in place a pipeline which is called Solution to Structure, or SOS pipeline, where we can give limited access to groups coming with a liquid sample, and we do a couple of tests to try to vitrify and obtain grids which are of sufficient quality to allow then high-end data collection using the Titan Krios microscopes. All the information, all the metadata and images are saved in ICE-by-V in our LIMS system, as I've shown before for this one as well, and that the microscope is of very good quality, shows that we have recently obtained a structure at atomic resolution of 1.63 angstroms. All right, these facilities, roughly and quickly going through, which we have as ancillary facilities, comprises, for example, spectroscopy, and you see that you have different modes of these spectroscopy. They're available both online and offline. And there's a nice paper from 2015, a little bit older, but showing you basically that with which of the spectroscopy you get additional information about whatever the functional state, kinetics, radiation damage of your sample. A very nice example was the mechanism of the urate oxidase, where people were discussing a mechanism based off the whether there's a hydroperoxide intermediate or not. And if you were collecting classically, so with a classical dose, you would only see this picture, which you see on the left, so an oxygen bound right above the cofactor. But if you check this one with spectroscopy, you see that there's something else going on, and you could confirm that there's this peroxy intermediate formed when going to very low dose data collection. Another ancillary facility which is quite interesting, or could be quite interesting for you, is our high pressure MX facility, where you can basically get your complexes cryoprotected without using cryoprotection. Also, you can do experiments with gases like oxygen or xenon, krypton, to see how these gases are entering and leaving your protein. All this one is only possible if you go to whatever 2000 bar or more, because then you go into the high density water phase, and this one you can directly freeze without any cryoprotection. Picture shows that it works. One nice thing that was actually a trypsin crystal with benzamidin, and from molecular simulation experiments, everybody thought that the benzamidin inhibitor comes in from this side, but we actually could populate a low binding site over here and could show that this is differently. And also this is a recent experiment in which you see that the krypton as mimicking oxygen, and you could show the path how oxygen enters into this protein. All these ones, because we want to go finally to this picture, we want to get more information. You have the structure only, but you want to have a little bit more information about dynamics and the function. If you combine different techniques using different ancillary techniques, you get, of course, more and more information, and this shows you a little bit more how the protein acts in real time. And with this one, I want to finish and thank you for your attention and open for questions if there are any.
Thank you very much, Christoph. That was an extremely interesting talk, and I learned a great deal about the facilities at ESRF, and it certainly has changed since we were regular users. In many respects, by teaming up with Diamond, we've missed on the developments that have been happening at ESRF, and I think we should certainly start utilizing the resources a bit more in the future.
Sounds good to me.
Well, I would love to. It's just it's a matter of money.
Yeah. Well, but you know, I mean, the ESRF is quite good in I mean, for the member countries, that's the transport, for example, is taken care of, and you have quite a lot of things that you can do within one trip or within one remote access experiment. I didn't talk about this one. Currently, due to the pandemic, of course, we're running in complete remote access.
Yes, that's a real disadvantage for us, and it also precludes the use of the sample preparation facilities that are at ESRF and PSB and
sample preparation is certainly an issue. We put in place a mail-in service even for the BioSAX beamline. So the samples, the preparation for the BioSAX beamline is being taken care of by our staff. The other beamlines, they run mainly either as mail-in experiments, so you define beforehand what you want to have done, or as remote access. That means that we put your samples into a sample changer, and then we give you control, and you perform your experiments as you like on the fly. But we all hope that this goes over this pandemic rather sooner than later.
Can I just ask the various African people from countries other than South Africa, has anybody had an opportunity to use the ESRF? Can people put up their hands and speak?
Well, certainly not a lot. That's certainly true.
Emmanuel, have you used the ESRF?
Yes, I have used the ESRF, but based here in Europe, so not like from Africa. Yeah, I mean, there is a lot of upgrade, but when I was there, I really like it very much. I solved my structure from there. So.
And Daruda, have you used the ESRF?
Hi, Trevor. So I am currently over there. So
yes, you're on site. You're the main man.
I'm on site actually. So yes. So
not even 100 meters away.
Yeah, exactly. Yeah. So well, obviously, so I've used the beam lines, and I am currently serving as a support scientist on the Titan Cryos. So I am happy to discuss with anyone who wishes to have more information on access to the Cryos and obviously other ancillary facilities, so such as the PSB and other things. So thank you.
Thank you, Daruda. Thank you. That's very good. Does anybody else have a question or a comment or a query or anything else?
I see in the chat that Andrea was went to ESRF to do high pressure Krypton data.
Good. Good. Well, anyway, I mean, if you have any questions or anybody, don't hesitate and send an email to us. We're happy to reply to any kind of question.
Okay, thank you very much, Christoph. That was
You're welcome.
Thank you.
Thank you.
Is it now me to talk?
Believe so.
It is your turn.
Okay. Okay, I'll introduce, well, I just have to introduce myself. Okay, can I share my screen? And where are... There we are. Okay, and now share. And now slideshow play from start. There we are. Okay, can everybody see that nice picture?
Yes.
Good. Okay, so I'm going to talk about a project that we're busy with at the moment, which is towards understanding the mechanism of the amidases. And you'll see as time progresses, this has been a really long-term, ongoing project. And it just shows how you can easily jump to conclusions based on inadequate data when you're talking about enzyme mechanism. And I would advise at this stage of my jaded experience that a great deal of caution is necessary in order to, before you actually make a statement about how enzymes work. So I'm going to, oops, no, why won't it change? Yes, there we are. Okay, so I'm going to talk about amidases of the nitrulase superfamily. And the amidase mechanism converts an amide into a carboxylic acid. And it definitely does pass through a thioester acyl enzyme intermediate. There is no doubt about that. And that has been observed both by mass spectroscopy and by X-ray crystallography in the case of the yeast omega amidase. So that's true. Very closely related enzymes are the nitrulases, and these convert a nitrile into a carboxylic acid. And the literature is full of the story that says it goes via two intermediates, the thioimidate and the thioester intermediate. Now, let it be said that there is evidence by mass spectroscopy of an intermediate, but there's no evidence of what it actually is. So what happens in this step here, the middle two steps that I've drawn here, is a complete black box in the case of the nitrulases. Then there is a curious one that occurs in fungal species, which is called the nitrile hydratase, and it takes nitriles and converts them to amides. So it seems to do perhaps half of the nitrulase mechanism. And that is supposed to go via a thioimidate. Again, there is absolutely no evidence for this at all. And yet the literature says this with great confidence. So let me just say something about the occurrence of these enzymes and why they're interesting to study. So they're ubiquitous enzymes, and they're used for multiple cellular processes for the on-site production of ammonia, generally from glutamine. Nitrolases are somewhat less common. They do occur in plants. They're used in the glycosylate breakdown pathway to convert beta-cyanualanine to aspartic acid and ammonia. And they also occur in many bacteria and some fungi. And nitrile hydrotases are actually quite rare enzymes. They occur in fungal plant pathogens as cyanide hydrotases. And some nitrolases have substrate-dependent nitrile hydrotase activity. So there is a close relationship between nitrolases and nitrile hydrotases. So other members of the superfamily have different but related chemistry, and for some, the chemistry is not known. So one of the comments that I'd like to make is that what we have is a very versatile active site, and minute tweakings of the active site can generate different catalysts, depending presumably on evolutionary serendipity. So what they look like is this. They have a very highly conserved fold, which has a layer of alpha helices, 2 layers of beta sheet, and another layer of alpha helices. And they have a long C-terminal tail, which we've now started discovering has a definite role in stabilizing the nitrolase structures. But I'm not going to mention, that's another lecture. So what is interesting is that in the active site pocket, there seem to be a small number of amino acids that are pretty highly conserved. And these include 2 glutamates, a lysine, which in most of the structures that have been solved at reasonable resolution is hydrogen bonded to those 2 glutamates. We have a tyrosine that is hydrogen bonded to one of the glutamates. We have often a water molecule which is hydrogen bonded to the 2 glutamates. And a little bit away, we have a cysteine. And the cysteine is the cysteine that ultimately forms the thioester in the case of the amidases. So we have a commonly conserved motif, which has obviously got quite a lot of catalytic potential. And we have a cysteine, which is involved in the catalysis in some way. Interestingly, the cysteine itself is located on a segment of 310 helix in a very strained configuration. It's very interesting, and I have no idea why that happens. Okay, so in 2007, I wrote a lot of rubbish, but this is what I proposed as the way that amidases work. This was at this stage just a working hypothesis to try and formulate my thoughts for the further investigation. So well, other people had previously proposed that a glutamate caused the cysteine to ionize from SH to S minus, and that the S minus does a nucleophilic attack on the carbonyl carbon of the amide, leading to a tetrahedral intermediate, which then became the thioester. And then what obviously then has to happen is that you have to have hydrolysis of the thioester. And at this stage, at the stage that I was drawing this diagram, I proposed that glutamate 142 was involved in the hydrolysis. And I set out to prove that, and I'll show you the little paper that we wrote, which tried to prove that. It turned out to be false. This glutamate catalyzed the nucleophilic attack on the carbonyl carbon of the thioester. And this then, of course, led to another tetrahedral intermediate, and that would then break down to release the acid product and restore the active site to its initial configuration. So the question then is, how does all this hold up? I mean, it's easy to draw a diagram like this, but you obviously have to have to visualize everything in 3 dimensions in order to figure out what's going on. So what happens is that the electron density in many of the amidases is quite well defined, and you can easily fit we've had amidases that have diffracted to about 0.75 angstroms, and some of them are remarkable diffractors. But in all cases, there is pollution of the active site occurring due to the oxidation of the cysteine. So this readily oxidizes to probably a mixture of sulfinic and sulfenic acid, and in that way confuses the interpretation of what is going on in the active site. So what we can definitely see is this motif here with the 2 glutamates, the lysine, and the water. And obviously we can't see the hydrogens by X-ray crystallography, but we can see enough to be able to predict that. And we cannot, in the case of the cysteine, it would be really handy to see where the hydrogen were located and to see whether there was any basis of the idea that in fact this cysteine is ionized by interaction with this glutamate over here. So it would be the people have chosen this glutamate to be the glue that ionizes this the cysteine. So the first experiment we did was we substituted the. You remember I told you that this glutamate was the one that was supposed to hydrolyze the thioester. And what we did was we substituted this glutamate with a leucine, and we found that we found that in fact a chloride ion from solution then sat in the position that the oxygen of the carboxyl was previously located. So it kind of confounded the experiment to some extent. The enzyme was obviously inactive, but we did discover quite an interesting thing, and that is that if we put in acrylamide, we formed this Michael addition with the acrylamide. So this is an acrylamide that is now attached itself to the cysteine and sits in the active site. And the leucine that we'd substituted the other glue with was sitting to the side, and there's the chloride ion. So we under the conditions that we used, we found no other adducts with the cysteine. And so what we did was we started looking at exactly what was happening here. And in fact, if you model this, you find that that the acrylamide actually alters its location relative to the cysteine. So in fact, the cysteine is now no longer sitting near the carbon on carbon, but it's sitting at this carbon. This is a double bond in acrylamide. It's sitting over here in this relative location to the acrylamide. And so what we get is a mechanism of Michael addition, which looks more or less like this, where you actually attack the double bond of the acrylamide to form this Michael adduct. Okay, so that was interesting. One of the reviewers said, well, what would happen if you put an aspartate in the place of that glutamate? And what happened was quite interesting, and gave some insight into the role of the D142. The enzyme was not active, but what happened was that we could clearly see that the aspartate carboxyl sat in two different locations, and so did the lysine. And in fact, molecular mechanics simulations of the system showed that it oscillated between these locations. Unfortunately, this Apple computer that I'm using now doesn't actually play this video, which is really irritating. But you can see the oscillation taking place between these two locations and the oscillation of the lysine taking place between those two locations, which is extremely interesting. So the active site is destabilized by substituting the natively occurring glutamate with an aspartate. And it seems that the destabilization is due to the fact that this location likes to be occupied by a negative charge, which is the same negative charge where the chloride ion sat. Same location where the chloride ion sat. So the problem that we encountered in trying to explore the mechanism in this way was that we got mispositioning, and it brought about a non-natural adduct. So we then decided that what we would do, and this was done by an extremely determined student from Kenya, Sarah Kimani, who did a huge amount of work in making all of these different substitutions, looking at the complexes that resulted from reaction with a number of different compounds and mostly substrates, and then trying to analyze ultimately the situation by X-ray crystallography. And so what we've done now is to look in the whole active site and try to put in the hydrogen positions, and we fill in the entire environment, and we try and use this as our starting position. Okay, this starting position has been obviously observed by other people. Now this is from a paper by Hay et al. and it's based on crystallography done by Hung et al. And one of the problems, I made a mistake here. This should be C166S. So the problem is it's based on a serine substitution for the cysteine. And the electron density that they had in the active site was extremely poor. But what they claimed to observe was a formamide, which you can see here, NCO, and these are the hydrogens, a formamide bound in between the two carboxylates of the two glutamates. So that was their starting position for the whole reaction. And they based a whole lot of quantum mechanical calculations on this starting position and created a mechanism in which supported the idea of an ionization. You can see here their cysteine is very close to the glutamate. They claim that this causes an ionization. Everything is correct in quantum mechanical terms. Trouble is that this starting location was so poorly observed that it can only be considered to be a fantasy. So one of the things that we've been trying to do recently is to try and undo this damage and look at really what the starting location looks like. And here, what we've done, this is done by Stanley Makamiri, is we've done in the cysteine to alanine mutant, we've docked in a whole lot of different amide substrates, and this is the average position of the amide substrate. And you can see that the carboxyl oxygen sits between the lysine and a backbone amide which we call the oxyanion hole, and the amino group of the amide is hydrogen bonded to a backbone carbonyl group and the glue, the one of the glutamates carboxyl groups. And if you then put the cysteine in position, so that was what happens when you put an adenine in that position. And if you put the cysteine in position, you don't get such a good fit. But that's almost expected, because what's going to happen is that during the reaction, you're going to form the thioester, and then part of the getting into that position is going to provide the energy for the reaction to take place. So here's what we observed, and you can see that the electron density is extremely well defined, and you can see that it's exactly as was predicted. You can see the hydrogen bonds between the lysine and the backbone amino group. You can see the hydrogen bonds all formed exactly as predicted by the docking experiment. So
Trevor, are you watching your own time?
No, you tell me how many minutes have I got?
Let's call it 3.
3. Okay, I'll wrap up. I'll tell you where we're going. So this is one of Sarah's experiments where she did the crystal structure of a propionamide, and this is now, you can see the propionamide sitting in the active site, and you can see that there's very good. Well, this is obviously the cysteine itself is modeled, so that's based on the adenine mutation, and you can see that there's very good stereoelectronic alignment over here. And that would then result in a thioester intermediate. And okay, so we have a lot of things to show that we have a lot of, okay, I won't go through that. But anyway, I'll show you what the main thrust of the current study is. You can see the docking is well done. You can see the mechanism by which we would form the thioester. And now we want to hydrolyze the thioester. And so what we did was a quantum mechanical calculation in which we showed the orbitals. And you can see that there's perfect stereoelectronic alignment between the LUMO on the thioester and the lone pair on the water. So you can see that this is all beautifully aligned, and the water molecule that does the hydrolysis is not hydrogen bonded to the glutamate. It's hydrogen bonded to the low numbered glutamate and the backbone, the same two that in fact enable the amide amino group to dock. So what we're showing is an absolutely beautiful illustration of how the hydrolysis occurs. And let me say that we didn't put anything into this. The water molecule was sort of floating around, and it went, no matter where we started off, it always went to this position. So this position is extremely well determined to bond to a water in this configuration. So we've proposed an amidase mechanism in which stereoelectronic alignment plays the major role, in which the catalysis is assisted by general acid and general base in the usual way. And the important new result is that it's not necessary to assist the cysteine in order to form the thioester intermediate, in other words, to remove the hydrogen. So I just want to thank the people that have played a role in this over many years. And the main players have been Brandon Weber, who's now unfortunately left the lab, and Gerard Venter and Roger Hunter, who are currently with us. JC Gumbart, who did a lot of the molecular dynamic simulations. Stanley, who's working on the project at the moment, and Sarah, who really broke the ice and got everything going. And of course, there've been lots of funders over the years. And these have included the National Research Foundation, the Robert Welch Foundation, the International Union of Crystallography, the Council for Scientific and Industrial Research, the University of Cape Town, the Deutsche Forschungsgemeinschaft, and recently the STFC GCRF START program. And those are all the logos for the people involved in the START program, which I have to show. So that's all I have to say, and thank you very much for listening.
So let's give Trevor a round of applause by the emoji at the bottom.
Well, thank you very much.
Emmanuel, do you we want to do some questions with Trevor before you start, or anybody have any questions with Trevor? Okay.
I want to make one point. This study has shown that you shouldn't believe what you read in the literature. The last thing that you should do is take the literature at face value. You should evaluate it, and not having the structural biology capacity to actually form your own opinions of other people's structural biology actually compromises your ability to actually use that data sensibly and in an intellectually satisfying manner. That's a very important point to actually tell our home funders, is that unless you're doing it, you actually can't just take it. And that is another thing that what I thought yesterday is that if we were dependent on other people doing our structures, and you know, you did a mail-in service, you got your structure out, and then you had to think about it afterwards on the basis of what they gave you, you would never have got as far as we did with this project, which required a lot of introspection and examination of the data at every point. Okay, that's maybe a good message.
Okay, thanks for that. So Emmanuel, or who's next actually? Trevor, who's next?
I am next.
Okay, gotcha.
Could you please give me permission to share my screen?
I think I've done that. Yeah, I think you can do that.
Okay.
Okay, now I've got to release my screen, haven't I?
Right, do that. Yep, there we go.
Right. So thank you everybody for coming to this talk. The title of my talk is Crystallizing Membrane Transport Proteins in Lipidic Cubic Phase, aided by an engineered green fluorescent protein thermal shift assay, which I call GFPTS. So throughout this presentation, I'm going to refer to the lipidic cubic phase as LCP. An introduction of myself. I did my undergraduate degree in Cameroon. Then I moved to Europe, did some master's studies, and I finally completed my PhD in Trinity College Dublin. So my research focuses on determining the structure of membrane transport proteins, and also to functionally characterize them. Together with Daouda and Mama, we involve in capacity building for structural biology in Africa. And I'm also involved in global health research to inform health policy on antimicrobial resistance. So my talk is going to be based on this paper, which we published in Network Communication. So I'm going to talk about just one aspect of the data. So some key facts about membrane proteins. They are essential for life, and 1/3 of the human genome codes for membrane proteins. They are important drug targets, and about 70% of all US FDA approved drugs are membrane proteins. And despite their pharmaceutical relevance, there are very limited structure of membrane proteins compared to that of their soluble protein counterpart. So the reason for this is because if you look at the membrane bilayer, this is an integral membrane protein. It's quite complex. You have a mixture of different lipids in the bilayer, and based on their function to transport their substrate from one side of the membrane to another, they are very dynamic. So you need a substrate to either keep them stable. So isolating them from the lipid bilayer, what you do is that you're removing the lipid. You're removing the lipids that are present in the membrane protein. So this makes them very, very unstable. And also, you have to use detergents, and the most common detergent, the detergent that is widely used is DDM. And the problem with detergent is that it forms a micelles that surrounds the membrane proteins, which makes it difficult to have that protein contact required for the formation of a well-ordered crystals. So once you purify your membrane proteins in the detergent environment, with the detergent, you need to crystallize them if you want to do X-ray crystallography. So for the first method you can use is the vapor diffusion, which is the standard method that most people would normally start with. So you need to use a robot to set up the crystallization trials. And in vapor diffusion, what you get, you see the crystals looking really, really nice in terms of the size and the shape. It's really nice. But what happens is that when you expose them to the beam, you see that they diffract really poorly. And the reason is that the crystals formed in vapor diffusion, they are called type 2 crystals. So you can see here, this is the crystal packing for this type of crystals. You can see a lot of channels here, solvent. So they have a high solvent content, that is water. So that makes them not to diffract very well. Unlike the lipidic cubic phase crystallization, well, what is the lipidic cubic phase crystallization? It's actually crystallizing your protein in a lipid environment. So what you do is you mix the protein on, you put the protein on one side of Hamilton syringe and you put lipid on the other side. Then you mix the two to form the cubic phase. So once you mix the two, and if you use SACs to determine the structure of that lipidic cubic phase, you will see different phases, which is dependent on the solvent content and temperature. So the most relevant is the cubic phase. So if you think about the cubic phase, this is what you should think of, like the toothpaste we use to brush our teeth. So it's highly viscous. So for the mechanism, once you mix the protein and the lipid, what happens is that the protein reconstitutes into the bilayer of the lipidic cubic phase. So once you add a precipitant solution in order to form a crystal, this precipitant solution perturbs the system, and as you can see, the protein migrate from the lipidic cubic phase to a local lamellar region. And once they locally concentrate, they form lipidic cubic phase crystals. And because of this shift of this lamellar shift, the crystals formed from this method are highly layered. They are very layered. So you can see the solvent content is very, very low. So they tend to diffract X-ray beam very well. So ideally, you want a crystal that will diffract the beam to high resolution for you to solve your structure. And clearly, the lipidic cubic phase has been the most preferred method for membrane proteins in terms of getting a high resolution structure. So to prepare the lipidic cubic phase, the benchmark lipid which we normally use is monoline. Most experts in the field, they thought maybe the monoline will mimic the lipids in the bilayer. That is, you're putting the protein back into the environment, which was the lipid environment. But actually, we observe that the proteins that have crystallized by the lipidic cubic phase are very, very stable proteins. For instance, the GPCR have been very, very successful, and one of the reasons is because they have high affinity ligands to keep them to lock them into one conformation so they can crystallize. So so before I move to the next part of my talk is the development of the GFP-TS assay, how we use it to crystallize two membrane transport proteins in the lipidic cubic phase. So before that, I'll give you a brief overview of the purification process. So if you have your membrane proteins, we normally fuse them with green fluorescent proteins. With green fluorescent protein, you can monitor the expression, you can monitor the purification process, and you can use that to measure the melting temperature. So you can do a lot of stuff using the GFP. So you isolate, after the expression, you isolate the membrane. So once you isolate the membranes, you need to solubilize the membrane with detergent. And with that, you can measure the TM, or you can do the ligand screen with the membrane solubilized fraction, or you can go into purification. After that, you can still measure the TM, and you can do lipid screen. And after cleavage with TEV protease in this region, you can further crystallize and determine the structure of your protein. So the GFP-TS method was derived from the fluorescence detection size exclusion chromatography thermal shift assay, which is called FSEC-TS. So in this method, what you do is once you solubilize your membrane proteins, you heat at different temperatures, as you see here. You heat at different temperatures, and then you inject the sample onto a size exclusion column. So what you get is you get the fluorescence peak at the different temperatures. You realize that at lower temperatures, you get higher fluorescence, but at higher temperature where you have more denatured proteins, you have lower fluorescence. So you plot the fluorescence intensity against the temperature. What you get is the melting temperature curve, which is an indication of how stable the protein is. So the higher the melting temperature, the more stable the protein is. So based on this method, we engineered the GFP-TS. And the difference between the two methods is that if you go back, we do not require this size exclusion chromatography step. What you need to do is to heat the sample, and then you add the detergent beta OG. So beta OG helps to precipitate aggregates that are in solution. So you can then use a plate reader to read the the fluorescence intensity, which if you plot, you can get, if you plot the fluorescence intensity and the temperature, you can get a melting curve. So if you see on the left-hand side, without the addition of beta OG, you can see aggregates, but in the presence of beta OG, all the aggregates in solutions are removed. So we measure the melting temperature for a cohort of bacterial proteins, as you can see here, using the FSEC-TS method and the GFP-TS method. And as you can see, the melting temperature correlates with no significant difference. So we were confident to say the method matches the gold standard, FSEC-TS. So for the applications with the GFP-TS, you can use it to measure TM, you can use it to measure affinity, and also you can do ligand screening. So, like I said, monoolin, which is the benchmark lipid used for lipidic cubic phase crystallization, most experts thought this lipid is stabilizing, but actually it's destabilizing. As you can see here for the sodium proton antiporter, if you look at the melting temperature if you look at the melting temperature, that's the control without any lipids, it's 42. And in the presence of cardiolipin, which we know it binds to this protein, it's actually 55. So cardiolipin binds to the protein and improves its stability. And you can see monoolin here is very, very destabilizing, the melting temperature dropping down to 27. So if you combine the two, cardiolipin and monoolin, you can see that it restores the stability of the protein and even improve it to some extent. So ideally, this is what you want to have a lipid that in the presence of monoolin is going to stabilize your protein to make it crystallize. So as you can see here, we set up the trials, we got lipidic cubic phase crystals, we exposed them, this was done at the ESRF, we exposed them and we got the crystals diffracting to 2.3 angstrom. So the last example is the nucleotide sugar transporter. This is actually CMP sialic acid transporter. So actually CMP sialic acid is synthesized in the nucleus, is then exported to the cytosol. It has to be transported to the Golgi lumen where glycosylation takes place. So this is the CST, the nucleotide sugar transporter that does that. And the decoration of secretory proteins with sialic acid is very important for some virus to get entry into their host. So in the initial process, we screen different nucleotides. And as you can see, CMP, which is the counters, after the sialic acid has been used for glycosylation, the remaining CMP is transported outside of the Golgi lumen to the cytosol. So as you can see, this ligand was very, very stabilizing for this protein. So we could measure the KD, which was somewhere around 37 micromolar. So we also screen the presence of monoolin. As you can see, monoolin was very destabilizing for the protein. And in the presence of CMP, you can see that the stability was restored to about 50 to 55 with or without monoolin. So we set up crystallization trials and we got crystals in the presence of CMP. And that these crystals diffract to 2.8 angstrom, and the structure was solved. This is a structure of the nucleotide CMP-sialic acid transporter. So we could use this method also to validate the residues that are coordinating the CMP. So once we mutate some of this residue, we realize that the stability reduces. That is a drop in melting temperature. So we could use the melting temperature before and after. We could measure the melting temperature for the wild type and the mutant and compare those. As you can see here, this correlates very well with what we are seeing in the structure. So overall, the GFPTS helped us to be able to crystallize these two transport proteins, and we were able to solve the structure. So that brings me to the end of my talk. I'm happy to take some questions.
Thanks, Emmanuel. That was a very interesting talk. Is the crystallography in these highly lipid-containing complexes, is it possible to get sufficient information to actually solve the problems that you're after? Are you able to get the information that you need out of the crystallographic experiment?
Yes, we were able. So the lipid doesn't have any interference. So once the crystals are formed, they are protein crystals. So sometimes you might have the lipid binding to the protein in some regions, but actually in this case we had the ligand the ligand was positioned where it's supposed to be, and we did mutagenesis studies to see that those residues actually are involved in the binding, and so we got all the information we wanted. And we did some biochemical characterization without lipids to get something like the KD of the protein to back up the crystallographic data.
That's interesting. Has anybody got any questions for Emmanuel? Okay, thank you then, Emmanuel. Thank you very much.
I think there is someone who wants to ask a question.
Yes, okay.
Hi, I'm John. I wanted to ask how common it is to detect electron density coming from the lipids. So do they stick to the external surface of a protein, or you also observe them in the core of the protein?
Yes, so the crystals is in the lipidic cubic phase. So once you collect the data, you have the lipid diffraction as well. So you will see that as a ring. If you saw you will see that like a ring. You
are able to resolve that electron density. You see them basically surrounding your protein.
Yeah, I think the software actually eliminates that diffraction from lipids. So it's been adapted such that doesn't interfere with the data processing.
So the electron density is subtracted. So it knows already how they look like, and it's automatically subtracted.
Yes.
Okay. Okay. Thanks.
No worries.
Thank you. I'm starting to get the hang of this now. I now know where to look for hands that are raised. So hopefully I'll get better at it by the end of the day. Okay, thank you for that question, and thanks, Emmanuel. So now we're at 11:22, and we have Dr. Mama Ndi from Umea University to talk to us about the structural basis of the interaction of chaperone CV3 with newly synthesized cytochrome during mitochondrial respiratory chain assembly. Mama, are you there?
I think I don't know. I'm meant to be next. Maybe I'm wrong.
Are you going to speak? Okay, so then there's something. Okay, so then go ahead. So sorry about that. The program says that she is going to speak.
Okay, I'm fine. I mean, maybe I'm looking.
Is that the topic? Did I get the topic right?
No.
I think we're behind schedule by 20 minutes. I think that's the problem.
Okay. Okay. So let me get back to the. Okay. Oh, I see. Okay. All right. We're behind schedule, and so you Gian, are going to talk about mining the PDB for tractable cases where X-ray crystallography combined with fragment screens can be used to systematically design protein-protein inhibitors.
Yeah, it's a very long. Yeah, basically, that is what I will try to talk.
Okay. Well, over to you, Gian. And as Lawrence said, we're 20 minutes behind, and so we should start 20 minutes late and end 20 minutes late.
Can you allow me to share the screen, please?
Yes, you have that. You should be able to do it right now.
All right.
Yep, beautiful.
So good morning, everyone. And so my name is John. I'm a research fellow at King's College. And in the next 10 minutes or so, I will try to show you how is it possible to combine X-ray crystallography and fragment screening to systematically design PPI inhibitors. Just to confirm, can everyone hear me?
Yes.
Okay.
Yes.
Perfect. So first of all, what is X-ray crystallography based fragment screening? So how does it work? So what you do, you basically crystallize your protein target in a 96 well plate. So you have your protein crystal in the well, in the drop. Then you use an acoustic dispenser to dispense in a single fragment onto the drop containing your crystallized protein. Then you take advantage of the soaking properties of a protein crystal. You wait some time, and then you pick the crystal and you check on, you shoot them at the synchrotron, and you check whether any of the fragments has bound onto your target area. So basically, hopefully at the end of the exercise, after you've done your screen, you will have identified some fragments that have bound to your target. And basically, informed by the structural information, you repeat this process again. You do this in an iterative way. So you go back, you redesign your fragments, you try to optimize the binding affinity of your fragment, and you repeat this exercise until you get to a point where you have a meaningful binding affinity of your fragment. So at that point, you have discovered and you have evolved the fragment into a ligand that can affect the property of the biochemical and biological properties of your protein. So until very recently, this approach has been used to develop the molecules that target the catalytic site of enzymes. What we wanted to do was to check whether we could use the same approach to target protein-protein interactions. So basically what we did a PDB screen for protein-protein interfaces that can be targeted by in-crystal fragment screening. So basically the question we asked is, are there cases in the PDB whereby your protein crystallizes in a lattice whereby substantial portion of the surfaces are exposed to the solvent, so you can use, you can probe these surfaces with fragments. And on top of that, are these exposed surfaces used by the protein to interact with our protein? So in our bioinformatic analysis, what we did basically a matching exercise. We basically overlapped the exposed surface of the protein in the lattice to non-interacting site of the same protein. And basically what we discovered is that there are several cases where you can use this approach. So basically, you can use X-ray crystallography and fragment screening in a medium throughput way to create protein-protein interaction inhibitor. So among the many cases that we identified, we decided to focus our attention on p38 alpha and interleukin-1 beta. So the reason why we chose these two proteins is because they are very well characterized. There are a lot of X-ray structures in the PDB. They crystallize in a lot of different lattices. And also it is well known which portion of the surface they use to interact with our protein. So at this point, we know that in theory, this approach could be used to develop protein-protein interaction, but then we wanted to test it. So the first thing we did, we crystallized interleukin-1 beta in the lattice that is described in the PDB code 2NBH. Basically, this it's a very nice lattice because when the way the protein interacts, crystallizes in the lattice, exposes substantial portion of the surface to the solvent. So as expected, we identified several fragments that bind to the surface of the protein. So basically, in a single exercise, we identified what is the chemical space worth exploring if you want to target specific interaction. So basically, you know what are the features, the chemical features a molecule needs to have if you want to bind in a specific site. For example, here, if you want to bind on this site 3, you need to have certain features that take advantage of a network of hydrogen bonding. Then the next things we did, we observed that these sites basically overlap with the binding sites of our protein. So basically what we observed is that site 1 and 3, they directly, so the ligand that bind in site 1 and 3, they directly compete with the receptor, whereas the ligand that bind in site 2, they directly compete with a therapeutic antibody that is called canakinumab. So then we did a second test case. So we studied p38 alpha. So in the lab, we have a long-standing interest in the p38 alpha. And basically, we crystallized p38 alpha in 2 lattices just to maximize the surface we could explore with fragment screening. And basically, in a single experiment, we basically identified all the known binding sites that had already been reported in the literature. So we identified lots of fragments that bind into the ATP binding site, and we could compare the features of these fragments with what is known, but what are the features that a binding site for an ATP site needs to have. We identified fragments that bind to the canonical site, to the tryptophan 197 pocket, and to the non-canonical site. So at this point, we wanted to kind of test, okay, by doing an X-ray based fragment screening, we can identify fragments that bind to the surface of the protein, but can we develop this fragment and improve their binding affinity so that they become stronger binder? So to do that, we decided to focus on the non-canonical site. This is a binding site that is used by p38 to interact with another protein that is called TAB1. So what we did, we said, okay, let's try to develop molecules that target this site based on the fragment we have seen in the screen. So we zoomed in onto this site, and basically what we observed is that we have 2 classes of fragments that bind into the non-canonical site. So there is a class of fragments, like several elaboration of adamantane cages, that bind in an hydrophobic pocket in the non-canonical site. And next to it, there are several phenylsuccinamide moieties that bind. So basically what we decided to do was to, based on the relative position of the 2 fragments, we decided to link the 2 and check whether by linking the 2 fragments, we could improve the binding affinity. So basically what we did, we introduced several linkers between the 2 fragments. And the end result is that while you cannot detect in solution the individual fragments, because they bind too weakly, these are basically probably millimolar binders based on the soaking concentration. Once you join the fragment together, you basically bring down the affinity into the micromolar range, and you are able to detect them by ITC. So basically the structural information, they inform you and they suggest you ways to link the two fragments together. So in this type of approach, we use an approach that is called the merging approach, whereby you have two neighboring fragments and you join them together to improve the binding affinity. So basically we had an improvement in binding affinity of several folds. Then, as you might be aware, the COVID-19 struck, and basically we decided to check whether we could use this approach also trying to design antiviral drug against SARS-CoV-2. So as you might be aware, the SARS-CoV-2 uses a domain on the spike protein that is called the receptor binding domain to enter into human cells. So basically, it interacts with the ACE2 receptor on human cells. So our idea, and this is just a new project that is just started in the lab, was we could basically stop the virus from entering into human cells if we can design molecules that bind onto the receptor binding domain and they directly compete with the receptor binding site. So in this way, we basically inhibit the interaction between the ACE2 receptor and the spike protein. So this is just early stage. So the first thing we did, we basically did a screening exercise to look for several type of lattices. So basically, and we wanted to find a lattice that could be used as a soaking system. And during the screen, we identified a crystal form that we think is ideal to do this type of exercise, because the RDB in the crystal form we have identified crystallizes in a way whereby it exposes completely the surface it uses to interact with ACE2 to the solvent. So basically, in the next slides here, I'm just trying to convey how wide and how big is the solvent channels facing into the ACE2 binding site of the RBD2 domain. In other words, basically here you can model all the residues that are responsible from ACE2, that are responsible for the interaction with the RBD without creating any steric hindrance, steric clash. And basically, I hope that I have convinced you that the combination of X-ray crystallography and fragment screening can be used to systematically design protein-protein interaction inhibitor. So I have presented to you 3 cases, like interleukin-1 beta, P38 alpha, and the receptor binding domain of SARS-CoV-2. But there are over 400 cases in the PDB that, at least in theory, are possible to be used with this approach. Finally, I want to thank all the people that have been involved with this work, the people from King's College in London, the people from the Scripps Institute, and the Diamond Synchrotron in Didcot. And finally, I would like to thank you for your attention. And if you have any question, I'm happy to answer any question.
Thanks, John. That was extremely interesting. And I'm very impressed to see how linking the fragments increase the affinity of the drug for the binding site. It's remarkable, and I guess it's not, physically not remarkable, but the fact that it was accomplished is remarkable.
It is the structural information. I mean, it's very difficult to link together two fragments, even when they are close. The room for error it's quite big. But if you have structural information, the advantage is that it minimizes the number of combinations you try, and it facilitates a lot the chemical effort.
Did you do that experiment at King's?
Yeah.
So you have a really hot synthetic chemistry group.
So this was done in collaboration between King's and the university in Ukraine.
Okay. Okay. Can I see? Are there any hands up? Can anybody see?
Yeah, can I ask a question?
Yes, of course.
Yeah, thanks for this very lovely presentation. So by the individual fragments, you had millimolar affinity. Once you link them, you have micromolar affinity, right? So what is your plans in terms of improving this affinity? Are you going to do some maybe chemical modification of some side groups and so on to see if it's going to improve?
That's basically the next step, and that is it's a little bit more difficult. So the end target, so the affinity you need to reach, depends on what is the endogenous substrate you are trying to displace. So let's say if the protein you are so basically what we are doing here, we are trying to design some molecule that would inhibit the interaction between two proteins. Now, if the interaction between these two proteins is in the nanomolar range, your fragment or your molecule needs to be stronger, and so it needs to be lower. It makes it more difficult. If instead the interaction is in the micromolar range, it's a bit easier. So in this specific case, what we are competing against is a protein called TAB1, and the endogenous binding affinity is in the micromolar range. So we are almost there. We are almost there in terms of competition, and we just need to improve it maybe tenfold. So the next steps is to do some smaller chemical modification and try to optimize the binding. But if you are competing with a binder that is in the picomolar range or nanomolar range, then it's much more difficult. And you might reconsider the type of fragment you have decided to avoid, because the difficult thing is that you have several fragment binding, and then you need to make a decision which one you want to carry forward and improve. And it's not always straightforward. It's that point where a difficult decision needs to be made. Did I answer your question?
Yes, very well.
Okay, thank you. We're now at 11:40.
Yeah. Elvis, yeah, I have a question. Oh, we are running late.
Elvis, okay,
Yes.
let's take your question then, and let's.
Okay. Yeah. So very quick. So you are looking at a domain. So what informed the design of the ligand? Because sometimes you need the geometry of the active site to be able to design a ligand towards that active site. But this domain is just a motif. So what drives the synthesis?
So we are not playing with domains. These are full-length protein.
Okay.
So we decided to focus on full-length protein. So these 400 cases are full-length protein. And basically, what drives the chemist is the chemical feasibility. So the fragment screening is done using fragments that are chemically tractable. So you don't want to be in a situation where you find a very nice fragment that is decent, but then you cannot do any chemical modification because it's very difficult to carry it forward. So it's a compromise between how easy it is to do chemistry on it and how well the fragment binds.
Okay. All right.
Okay. Thanks, John. Thanks, Elvis.
Yeah.
Okay. Now we're at 11:42, so we'll aim at stopping just after 12. And it's Mama Ndi who's going to speak to us about the structural basis of interaction of the chaperone and CBP3 with newly synthesized cytochrome during mitochondrial respiratory chain assembly. Mama, it's over to you. Can you share your screen?
Yeah, please, can you give me permission to share my screen, please? Thanks.
Are you there, Lawrence? Are you giving him permission to share his screen?
Yep, he's got it now. Sorry, I was a little.
You were sleeping?
No, I was multitasking.
Okay.
You should have it now.
Okay.
Okay.
There you go. Sorry to be a few minutes.
Okay, hello everyone. Mamanzi, postdoc fellow in Department of Medical Biochemistry and Biophysics, Unyo University, and one of the co-founders of BioStruct Africa. So I'll talk in two phases. The first phase will be on structural basis of CBP3 interaction with newly synthesized cytochrome B during mitochondrial respiratory chain assembly. And the second part, which is my ongoing project, will be on expression, purification, and crystallization of a G quadruplex DNA-specific antibody, BG4. So I'll give you a brief introduction of the projects and then followed by rationale and objective of the study. I'll summarize major findings. I'll give you conclusions and future plans, and then I'll round up with acknowledgement. So the first part of my talk. So the energy from the food that we consume end up as electrons, and they are carried by reduced metabolites to the electron transport chain located in the inner mitochondrial membrane. At the electron transport chain, the electrons are transferred sequentially through the respiratory chain complexes to the ultimate electron acceptor, oxygen, which is reduced to water. At 3 of these respiratory chain complexes, complex 1, 3, and 4, the transfer of electrons is coupled to the translocation of protons across the inner mitochondrial membrane, generating a proton motive force that drives ATP synthesis via respiratory chain complex 5, also known as ATP synthase. So this molecular machine accounts for about 85% of total ATP that's generated in the cell. And that justifies why the mitochondria is often described as the powerhouse of the cell. The subunit of respiratory chain complexes are of 2 genomic origins. You have those that are nuclear encoded and they are transported from the cytoplasm into the mitochondria, where they are incorporated into the respiratory chain complexes, and those that are mitochondrially encoded. So what you see, it's actually a sequence of events that takes place in the early stage of biogenesis of respiratory chain complex 3, which is a central component of the respiratory chain. Now, the protein CBP3/6 complex is involved in a feedback loop that coordinates translation and assembly of respiratory chain complex 3. So if this CBP3/6 complex is absent, then the assembly of respiratory chain complex 3 will be disrupted with far-reaching consequences. Unfortunately, we do not actually know how this CBP3/6 complex accomplishes this task, and that's partly due to lack of structural information in the protein. That brings me to the objective of this first study, which is to determine the structure of CBP3 and identify its interaction surfaces with CBP6 and cytochrome B. So we started with in silico analysis of CBP3, where 2 domains were identified. You have a his-specific, mitochondria-specific N-terminal domain and a more conserved ubiquinous cytochrome C chaperone domain. We look at the effect of deletion of cytochrome B, and our result shows that we need intact CBP3 for efficient respiration. We also look at the stabilization of cytochrome of CBP6 by CBP3. And our result here, as you can see, shows that we need intact CBP3 for stabilization of CBP6. So our next stage was to actually determine the structure of CBP3. So we screened several homologs, both prokaryotic and eukaryotic, and it turned out that CBP3 from Brucella abortus was the best candidate, and its structure was resolved to 1.4 Armstrong, and it revealed an all alpha helical unique fold with the N-terminal domain, the N-terminal helix flipped away from the rest of the helical bundle. 2 bound magnesium ions were also identified, and the first site being coordinated by these residues. And see the second magnesium binding site coordinated by these other residues. We determine the surface potential of the structure, and it shows that you can see colored from positive in red and to negative in blue, and shows that the protein, the surface of the protein is highly charged, which means its interaction may involve electrostatic interactions. So we use this crystal structure as a template to model CVP3 from Saccharomyces cerevisiae. You can see the model of CVP3 from Saccharomyces cerevisiae superimposed with the crystal structure. Here you see the superimposed model and the crystal structure, but with strictly conserved residues highlighted in yellow. Also determine the surface potential of the model, and it shows that it's also highly charged with it's colored from positive in red to negative in blue. So the next task was to extract information from the crystal structure and the model, put together and see how we can identify the interaction surfaces of CVP3 with CVP6 and cytochrome B. And for that, make use of a method that I established during the first year of my PhD, which is known as site-specific photo cross-linking. So this method makes use of a photoreactive unnatural amino acid that comes together with its orthogonal synthetase and its orthogonal tRNA. So the synthetase charges the tRNA with this orthogonal photoreactive amino acid, and the final result is that the photoreactive amino acid is incorporated at a specified position in this targeted protein. This is actually examples of photoreactive amino acids that can be used with this approach, but we use PBPA for our research. This is the workflow of the procedure, and by comparing sample in the presence and absence of PBA, we can determine if there is interaction at for a given position. As you can see, position R245 and R245. R245. You see that it forms a cross-link product. So in that way, we screen positions on the surface of the structure, and we selected this position based on conservation and exposure. So these are cross-link products formed at various positions. You see purification of cross-link product from a specific position, and probing with anti-CVP3 and CVP6. For instance, if you see this cross-link product form at this position, it's recognized by CVP3 antibody and also by CVP6 antibody, suggesting that it's a cross-link between CVP6 and CVP3. So in that way, we map other, we screen other positions, and we are able to map the surface of CVP3 that interacts with CVP6. In the like manner, we also screen for CVP3 interaction with cytochrome B. And for this position, for example, we see a cross-link product that is recognized by both CVP3 antibody and cytochrome B. To confirm this, we inhibited cytosolic translation and then labeled mitochondrial translation so that we have cytochrome B labeled. And then in that case, if what we see here is indeed cytochrome B, and then we should be able to see we see this in our autoradiography. Using that approach, we screen all the positions for interactions with CBP, with cytochrome B, and we can see positions that actually have signal for cytochrome B in complex with CBP3. In that way, we were able to map the surface of CBP3 that interact with cytochrome B. So putting our structural and biochemical data together, it came up with an interaction model for CBP3. So CBP6 binds at this region of CBP3, while the N-terminal domain, which didn't show any interaction with CBP6 and cytochrome D, it kind of binds to the mitoribosome and positions this complex within the tunnel exit for efficient recognition of a substrate as it emerges from the tunnel exit. So we're able to solve the atomic structure of ubiquinous cytochrome C chaperone domain from Brucella bottles. And we have mapped the interaction site of CBP3 with CBP6 and cytochrome B. And our data also suggests that CBP3 contacts the mitoribosome via its N-terminal domain. So the second part of my talk is on expression, purification, and crystallization of G quadruplex DNA-specific antibody, which is ongoing. So for some reason, I'll keep details about this talk. So G quadruplexes are non-canonical DNA structure formed by guanine-rich DNA strands. It was first detected in vivo in ciliates when G quadruplex specific antibody were used to stain telomeric G-force. And subsequently, the same approach was used to visualize G quadruplexes in human cells and cancer tissues. And it was actually observed that G4 foci co-localized with human telomeres. More than 700,000 putative G4 sites have been identified in the human genome, and they are mostly located in the telomeres, promoters of oncogenes, and in 5' untranslated regions. So G quadruplexes are formed from stacking of lower structures known as G quartets. A quartet is indeed a planar array of 4 guanines stabilized via pairwise hydrogen bondings with often a central coordinating monovalent cation. And depending on the orientations of the glycosidic bonds of the guanines in the quartets, G quadruplex might assume either a parallel, antiparallel, or hybrid conformation. And when just a single molecule is involved, as in these cases, it is described as intramolecular. But when it involves more than one molecule, as in these cases, is described as intermolecular. So these structures are associated with a number of genome functions, like in telomere maintenance. They're also involved in genome and epigenetic stability. They're also associated with DNA replication and transcription. So G4 is a single chain variable fragment antibody that is specific against G quadruplexes, and it shows no preference for any particular topology of G quadruplex, and it has high affinity for intra and intermolecular DNA G quadruplexes, and it has been used to detect and map nuclear G4 structures in vivo. It also has been used to show that there is elevated level of G4 structure in cancer compared to human cells. So the specificity of BG4 has actually been used extensively to improve our understanding of the formation, distribution, and physiological relevance of G quadruplexes. However, it is not yet understood how the BG4 G quadruplex interactions occur. We think investigating fine details of this interaction in structural and mechanistic perspective would shed more light on the intrinsic properties of these structures in the cell. So our first objective is to determine the atomic structure of BG4 bound and unbound to DNA G quadruplex, and also to characterize the binding. Is how a construct looks like. It's actually a variable region of the heavy chain linked to the variable region of the light chain via a linker peptide cloned into this vector under the T7 promoter. So after optimizing expression, we're able to purify BG4, analyze it on the gel, and compare it with commercial form of the antibody, and it seems to be a purified BG4 seems to be intact and of good purity. We did a second level of quality control where we use immunocytochemistry to determine in vivo binding of purified BG4 compared to the commercial. And analysis of the results shows that purified BG4 binds to G quadruplex DNA comparable to the commercial one. So the next stage was to, now we know that our purified BG4 actually binds to, it's of good purity, and it binds to substrate. So we set up a co-crystallization trial of the antibody and its substrate. We obtained some crystals, which we have actually harvested, and we are waiting for synchrotron time. We should. We also assess the in vitro DNA binding properties of the purified BG4. And for that, we use a DNA template of mitochondrial DNA template that's predicted to form G quadruplex. And the control is a modified version of the template that doesn't fold to form G quadruplex. You can see the difference in size after folding. Upon addition of BG4, we see binding of the G quadruplex forming template. But we see the complex stuck in the well. So we tried maybe lowering the concentration of BG4, we could have the complex migrate into the gel. We did that. We still see binding at lower concentration. But also of interest, we noticed that this particular template forms both intermolecular and intramolecular G quadruplexes. So we think it's better to screen condition that favors just one form of the G quadruplex. And we're aiming at getting a condition that will favor intramolecular G quadruplexes. For that, we screen salt and template concentration, and we see that in sodium chloride, only intramolecular G quadruplexes are formed. And then we actually adopted this condition, which seems to be the best for future folding experiments. So we repeated the experiment again now with this new condition, and with 10% gel. In 10% gel, we could actually see binding of intramolecular G quadruplexes. While at 5% gels, we can see binding of intermolecular G quadruplexes to purified proteins. So far, we think we have established a protocol of BG4 production, and also we show that we are doing BG4 G quadruplex co-crystallization trials, and our result also binding studies also show that BG4 binds to G quadruplex specifically. So after the structure, we'll move on to generate point mutants of residues involved in binding, and we'll also aim to identify suitable position on the structure where we can incorporate photoreactive fluorescent amino acid for in vivo studies, and we'll also attempt to modify and improve the binding efficiency of the antibody. With that, I would like to thank my supervisor for Martin Ott for doing my PhD, and Sho, who is my current supervisor for the second part of the work. Thank you very much for listening.
Thank you, Mama. That was an extremely interesting talk, and it shows how complicated the field of structural biology is and how terribly important it is to do the background functional work and to analyze that in preparing your structures. I really wish you good luck in getting your structural information. It looks like you're well on the way, though. It's very impressive work.
Thank you.
Are there any questions? Oh, yes, there's a question. Let me see who's asking it.
Hi, I work with helicases, and we started analyzing the interactions between helicases and G quadruplex. And what we find is that we try a different, choice of different G quadruplex, which based on the, NMR or crystal structures are parallel, anti-parallel, with a variety of conformation. And we find, surprisingly, quite a lot of specificity. So we have a helicase that binds very tightly to one G quadruplex and nothing at all to another one. So I was wondering whether your antibodies display specificity, or whether this is something which, might need to be addressed.
For now, what I see with my antibody is that it binds to both intra and intermolecular G quadruplexes. And concerning specificity, I think we have used template that form parallel G quadruplexes, and also there are a lot of templates that we are still trying to characterize at the moment. So for now, I think that's still very kind of open to debate.
Thank you.
Thanks, Sylvia. You're up next. You know this.
Right. So, I didn't send you a prompt earlier this morning, Sylvia, but you wanted to take a few minutes to talk about your fellowship program? You need to unmute. Sylvia, you need to unmute.
Sylvia's there. You are. Oh, you're still muted. Sylvia, you're still muted.
Sylvia, still muted.
Okay,
Unmute yourself.
Sorry, I didn't work. I just want to, very quickly let everybody know of possibility of fellowships. So if there is, literally less than 5 minutes, I can show you just a small handful of slides to illustrate these possibilities.
So go ahead and share your screen, Sylvia.
Hold on a second. I need to make sure she can do that. Okay, go ahead. You can share your screen now.
Okay, so the occasion for that, in Trieste, there are a number of international institutions that offer fellowships and opportunity to scientists coming from all over the world. And just very recently, the Elettra Synchrotron Facility and ICGB team up to offer a new scheme, which are fellowship, which are 2 years fellowship for scientists, young scientists and postdoc coming from all the countries which are member of the ICGB consortium, to work at the interface between Elettra and ICGB. So the fellowship will be for 2 years, renewable for a 3rd year, and will need to be endorsed by an Elettra scientist and an ICGB scientist. Just to give you an idea, these are all the countries which are member of ICGB, and the ICGB partner can be in Trieste, but also New Delhi or Cape Town, which are the main site of the ICGB research centers. And I can just provide the, just rather than describing in detail, I can actually send to the organizer this presentation so people can have a look. But just looking on the website, the research at ICGB covers infectious diseases, non-communicable diseases, medical and industrial and plant biotechnology. And just to remind you about Elettra, you heard about ESRF this morning. I just want to stress that Elettra is not a synchrotron which offers all the opportunities of ESRF. For example, there are no Microsoft beamline, but it is a good run-of-the-mill synchrotron, and especially there is a new, I'll just go quickly, there is a new beamline dedicated to macromolecular crystallography that works very well, and that offers also remote data collection. So again, for African scientists, this can be a possibility, and you could contact Annie Hereux, which is the leader of the beamline. So just to finish, I really don't want to take much time. I want everybody to be aware of all the opportunity that the Trieste system can offer. And beside the Elettra ICGB Fellowship that I just mentioned, the International Center for Theoretical Physics also offers some opportunities in biophysics, which includes many school and workshop in Trieste and abroad. Years ago, we had a biophysics school in Cape Town, which was organized also with the help of Trevor. There are fellowship for PhD student or fellowship for postdoc that can work across different laboratories. For example, the STEP fellowships allow students from developing countries to spend 3-4 months a year in another laboratory abroad. And there is also the Organization for Women in Science from Developing Countries, which offers not only South-to-South fellowship, so a student or a postdoc from Malaysia, can work in South Africa, or someone from Zimbabwe can go to Brazil, but also startup grants for young investigators coming from science and technology lagging countries. And I stop here. I know lunch is looming and I don't want to take any extra time, but if anyone wants to know more about these, they can send me an email and I can put them in touch with the relevant people or institutions.
Thank you very much, Sylvia. That was very useful, and I hope that people take you up on your offers. Are there any questions for Sylvia specifically? Let me see if any hands are up. And I don't see any hands up. Ah, there we are. Elvis wants it too.
Yes. Sylvia, this is very good information. But I see that the Elettra, they say Ghana pending. What does that mean?
Elettra, sorry, say it again.
They say Ghana is pending when you show the map.
Yeah.
Does it mean that we are not there yet?
I think there are countries which are already full members and countries which are in
this one
to join, for example. So I know that, for example, Zimbabwe, that was used to be one of the pending countries, has recently become a full member. If you want to, I don't know, help.
So I'm speaking specifically to Ghana.
Again, yeah, I mean, I can put you in touch with the head of ICGB, which is to involve more countries, and possibly you may know someone that can help this process.
All right, thank you.
The leadership of ICGB is extremely open to enlarge the pool of countries which are members. So I work at Elettra, not at ICGB, but I have a number of contacts, and I can possibly, if you send me an email, I can put you in touch with the head of ICGB, and I know they are keen to have more contacts and having more people interesting.
Okay, thank you.
Are there any other people that want to ask questions? I don't see any hands at the moment. Okay, with that, I'll close this morning's session. And then, Lawrence, have you got the program up to see what time we're reconvening? It's at 3 o'clock, is it?
1500 GMT +2.
It's 3 o'clock South African time.
Yep.
Yeah. Okay. So that's when we're reconvening. I will wait here. Let's keep the session open during lunch. I believe that somebody, Roberta, wants to come and test her slides.
Okay.
During lunch break, and so I'll sit here and wait for her to come in, and we can do it whenever she's ready.
Okay, we'll do.
I'm here. Can you hear me?
Yep.
Hi.
This is Roberta.
Yeah, you can test. We can break for lunch, and I will allow you to do the settings. So you can do your screen sharing. Okay. So all right. See everyone at 1500 GMT +2.
Okay. Thank you.
All right. Thanks.
Thanks to all the speakers of this morning, and thank you very much for everybody that was in the audience. I think it was a great session, and I really enjoyed it. Thank you.
Ciao Silvia.
Ciao.
Hi, Sylvia. Thank you very much.
You're very welcome.
Do you like this way of doing things? It makes people much more accessible, you know.
Yes, it's true, actually. I mean, it would have been a lot more difficult to travel. There is a lot more opportunities to interact.
Yeah, I think it's fine. I think, and I think for our young people, it's a remarkable opportunity to meet all the people around the world.
Yeah.
And I mean, it's so difficult. You go to a conference, you don't actually meet the people. And this medium actually exposes the people to you in a very cost-effective and helpful manner.
Yeah. No, and especially the cost effective. I mean, I have people in my lab which are, normally to go to a meeting, you spend quite a lot of money, so you need to be selective. And now, because the costs have become much lower, they have many more opportunities to hear good people talking and chat. So it's something we need to somehow keep. I mean, you do miss the one-to-one, the coffee breaks and the more informal chatting that you can have, but it is it's a parallel opportunity that we should probably keep up.
I think so.
I would have loved to go to Cape Town anyway.
Yeah, no, I mean, yeah, that will be nice. That's true.
Well, you must come when it's possible. I don't know when it is going to become possible, though.
Believe me, as soon as it becomes possible, I'm going to travel like a yo-yo. I miss it.
You miss a lot, huh?
I feel so stuck, I was also used to go to London reasonably often. I've got a lot of friends. Now I feel completely trapped. So I'm going to spend 2 months holidaying all over the world.
Okay, Roberta, when do you want to do your slides?
Oh, and even now, if you like, if you want, I'm ready.
You can go ahead.
See you later.
Okay, then. Ciao, Silvia.
Ciao.
Okay, then I'll just share my screen, I guess. Yes, I think this is it. Can you see the screen?
Yes, I can.
So, okay, then there is not much there are no animations and other stuff, so we can, I think it's fine. Do you see the normal view, the presenter view, or the other one?
Well, I can see your full screen and I can see your face. So
perfect. No, fine. So, We can go for lunch.
Let's go for lunch. Okay.
Okay, thank you very much. I should
Thanks, Chris.
also the audio is okay because sometimes I have problems. Hello?
Okay, I'm there. I'm here again.
I cannot echo, stop share. Here I am. Okay, thank you.
Okay, you can stop share, and then so the first person to speak is going to be Zoe Fisher, and then you're going to be the third person to speak.
Yes.
And the schedule time, if we don't run terribly far behind, is 16:20.
Okay, that my time is 3:20. Okay.
Okay.
We are 1 hour earlier, I think. Yeah.
Okay. Okay then. Thank
Okay,
you.
so see you later.
See you later. Okay then.
Thank you very much.
Bye. Clifford, are you there?
Yes, Prof, I'm here.
Just have a bash at sharing your screen. Turn on your camera and let me see you. Okay. You want to now can you share your screen? Have you got a green button at the bottom?
Yes, let me try now, Prof.
What's happening?
Still trying to locate it, Prof.
Okay, so if you it's a green button right in the middle at the bottom.
Yes, I did click on that green button, and I'm seeing this screen where it's asking for a basic, advanced, and files. So,
Okay, then.
So
and then if you say,
my files. Okay, you can go ahead.
and you see a whole lot of windows, of all the windows that are open on your computer. Do you see that?
Yes, I can see like Dropbox, Microsoft, OneDrive, Google Drive. Yeah.
Pass over till we find your window of your presentation. And then you click on that.
Yeah. Oh, okay. Because my presentation is on my desktop.
Okay, but you have to open your presentation so that it's in a window.
Okay, let me do that.
And then when you mouse over the window that you want to share, it has a little blue border put around it. And then you just leave that highlighted. You click on it, the blue border gets wider, and then you go down to the bottom right and you click share.
So actually I had some connection problems, but I'm back now. So I can't access the green button anymore.
You can't see the green button anymore.
I can see, but I can't access it. It says host disable participant screen sharing.
Host disabled participant screen. Okay, so let's go to the host. Let me Skype the host.
I'm here.
Are you there? Okay. So will you let Clifford do his screen sharing?
He should have that already. I mean,
Well, he says it says host disabled participant screen sharing.
all right, let me try. Okay, one more time. Try it again now.
Oh, all is good.
There you go.
It's starting to work. There you are. Well done.
Thanks, Prof. Excellent.
Okay, so then we'll just do that again, and I can hear you. It'd be nice if you got a light in front of your face, hey? You're very dark.
Okay. I'll try to adjust my view, Prof.
Okay.
Maybe the setup and our lighting system is a bit dodgy.
Your lighting system is a bit dodgy. Oh, you're in the lab.
No, I'm in our office area, sitting area, Prof. It's not a lab.
That came a bit better. Is there not a desk lamp in the office somewhere?
I think I know what the problem is, because I have my sitting space that is blocking the light. I'm just going to move away a bit, then we'll see. Just give me a second.
There you are, very good.
Excellent. Okay, so it was just a sitting position.
A little bit better a moment ago. That's the place. That's the sweet spot. So you get the equivalent amount of light shining on your face, then it's perfect.
Okay, yeah. Otherwise,
you just one complete even black tone.
Okay. Otherwise, the background is fine, Prof.
The background is just the white wall. That's fine, and but you darkly contrasted against it, which is I mean, it's not so helpful. You can't see your face or anything. There we are. That's the sweet spot again. So there is a sweet spot where you get a very nice view of your face. The light on you is perfect now.
Okay, I will have to see it. I'll have to make a plan. I have to put something.
Maybe you can look in Bob Dieter's office and see if there's a desk lamp.
Yes. Okay, Prof. I will sort that out before my presentation, perhaps.
Okay.
Thanks, Prof.
Okay, thanks.
Hello, there's no audio on my end. I hope I'm not cut short.
Can you hear us? Can you hear me?
Okay, I can hear you now. Okay, all right.
All right. Yeah, no one was saying anything.
Just waiting until 3 o'clock South African time.
Trevor, are you there? Trevor, did you see the email I sent you?
Am I on? Let me see. Let me have a look. Did you see me come there? I was out of the room making a cup of coffee.
Right. Then you came back onto the beach.
Then I came back on the beach. Okay, where's your email?
Why don't you? Why is your background like Table Mountain? Look like you have a desk on Table Mountain or something like that.
I've got the whole lot of Cape Town tourism pictures that I can use. I just like the beach. The beach is also fun, you must admit. I mean, having the waves breaking and the palm tree rustling is quite soothing, isn't it?
Yeah, for sure.
Okay, so this is the one about the survey. The survey
Right.
for. So you want me to fill it in?
I want you to see if it's if the questions are reasonable and whatnot. We haven't released it yet.
Oh, okay.
So what do you think? Looks reasonable?
Yeah, I think it's reasonable. I'm just answering the questions, and you can see what I think, and then you can see whether it's producing the required result. Can you see the answers that I'm giving?
No, they go into a. I think I have to stop the survey in order to. Let me see what's. I forgot how to see the.
Anyway, the point is that the questions are easy to answer. And I mean, they certainly test the water on a number of issues. And I think it's worthwhile answering it. And I think you've given other fields where people can put their own ideas. I think that's all fine.
All right, let me see. I'll see what Chart and Emmanuel think, and then I'll probably send it out either right after this session or before tomorrow's morning session.
Yeah.
I guess if people think the sessions are going well, we have until tomorrow. We have tomorrow and Friday to screw that up.
Yes. Shame, Lawrence. I think you're doing just fine, Lawrence. I think this is a wonderful idea, and I think you've shown us that there's a lot to keep us together.
Yep. For sure. So I was just reading in the news about the South African Airways airplane takeoff situation.
Oh, it's pathetic. It's so pathetic.
That is not cool.
No.
I always liked South African Airways, to tell you the truth.
Well, it was a very friendly airline, but unfortunately it didn't make any money.
Yeah.
Oh, no, alternatively, no, it probably did make money, but the people stole the money.
Well, Ethiopia has a brand new airport, and
I've been
also a
there.
very friendly airline.
Yeah, it's getting less chaotic, which is good. I mean, in the old days, the first time I flew on it, it was so chaotic. It was wild.
At the airport there in Addis?
And on the planes and everything. It was just a complete free-for-all.
Yeah, the airport in Addis needs, I think they try to jam in too much retail and not enough space for person comfort.
Yes.
But it's I haven't been to the new airport yet. So the airport in Ghana was very nice. Actually,
we have a modern airport actually.
Yeah, in Ghana, I deplaned. I went to the restroom, made passport control, and by the time I got to baggage claim, my bags and everything were already there.
Oh wow! When was this?
2019.
2009. Oh, just recently. Yeah.
But the thing in Ghana is that so for U.S. persons, we need to get a visa before we go. And
Yes.
I hate that process. I don't like giving up my passport and hoping that they'll get it back to you in time. And well, luckily I live in Washington, D.C., where, all I have to do is go to the embassy.
Yeah.
But when I was there was a gentleman who was from Detroit, actually. So he flew from Detroit to DC just to make sure that he can get his visa. But then, having gotten the visa, paid $100 for it, when I get to passport control, the guy just glances at it. And I was like, what did my $100 just get there? Because if it's the $100 is what the government wanted, I could have easily, I don't know, like, well, let's meet at the restaurant outside the and we could buy $100 worth of food and drink.
Yeah.
Well, what about the British who charge over £1,000?
Yeah, what is that about?
To get a visa?
Yeah.
That's just a tax.
Wow.
That's a tax on tourism, and it's counterproductive.
Yeah.
I told this people in at the Ghanaian embassy here in DC that well, back then the South African Airways was the way to get directly to Accra. And like on Friday, if there were seats available, there was like a number that you can call to get the deeply discounted ticket. And it's only like a 7-hour flight from here, so it's like it's shorter to get to Ghana than it is to get to Los Angeles.
Yeah, that's right.
So I would easily like take a 4-day weekend to Accra if I didn't have to get a visa, or if I could at least do visa on arrival.
Yeah, they used to do that actually.
Yeah. So was it 2019 was the year of return or something like that?
Yes. Yes.
Yeah. So I told the people at the embassy like nothing says return like visa on arrival.
Well, it's just that the Americans got dodgy.
Yeah. Right. Let me tell you my other story. So we were in 2019 with the African Light Source Conference. We were in the science minister's office, and we were talking about either him or the president visiting either ESRF or NSLS2 at Brookhaven. So I was talking about, so this is how you get from JFK to NLS2, that it's an easy drive, whatever. And the minister and his assistant started laughing. Turns out that day, I hadn't seen the news, but that day, the Trump administration had canceled all the B1 visas for Ghanaian government officials. Because, Trump administration was like xenophobic and racist. So there were all these people who these so-called Ghanaians who the government wanted to deport.
Oh yeah, I heard that. Yes.
And I guess it's up to the receiving country to pay for the airline charges. And Ghana did not want to pay for people on the list because they said they weren't sure that the people were actually Ghanaian, because they thought the list might have contained some Nigerians, blah 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 blah. Anyway, so that because the B1 visas have been canceled, none of the ministers or anybody could contemplate going to America, coming to America.
Yeah.
So it was like very embarrassing for me. I was trying to convince them to come to the United States to go to Brookhaven National Lab.
Yeah.
So, but the interesting thing is, as a group, Ghanaian immigrants to the United States have the highest incomes and mobility rates. So like, they may come here and get into like, medium scale jobs, but they quickly climb up, or they come here with very high skills and command very high salaries. So the United States actually, instead of deporting Ghanaian immigrants, should be getting down on their hands and knees and saying, thank you for coming here.
Well, I guess those didn't have a valid permit to stay in the United States.
Yeah, that's paperwork. It's like, you don't have your permit's not valid. Here's another valid permit. Maybe, I don't know, charge $50 for it or something like that. Or the thing about immigration is you can become legal or illegal at the stroke of a pen. So I always think that people should just be able to travel across borders at will. This should be like universal passports.
Oh, that would be dangerous. What is happening in the South now? People wanting to come to the United States.
Right. Well, you always want to be the country that people want to come to instead of get out of. So I find that very much not a problem. Trevor, wouldn't you want the best biophysics students in the world to try like hell to get into South Africa?
How would you attract them, Lawrence?
Money.
That's a good question, right?
Money solves.
Yes, and resources and facilities.
Right. So that's all money. That's all.
Established knowledge base. So it's all got to happen. It can't happen like by turning on a switch, right?
Yeah,
Mhm.
sure.
Mhm.
Yeah. So Trevor, here's a question for you. They're talking about, I just saw a story about how the new identified strains are more infectious, is the term that the media is using. So
Yeah.
what does more infectious mean from a molecular thermodynamics or kinetic standpoint?
Well, we think, just hang on, there's a message from John Rubenstein. Okay, I'll just say it. Okay, so he says he's going to, what is 915 ET?
That's 1515. He wants to start 1515 late.
No, he's going to log in on 1515. I just
All right,
said
he's at 40?
Toronto. He's at 940, yeah.
Okay. So you think, do these mutations sort of make the binding tighter?
Yes, it's the 501Y makes the binding 2.5 kilocalories per mole stronger.
Well, let me write that down. I'm gonna put that on Twitter. Is that a result that you have?
Yeah, I think it's published.
It's published. Okay. Which variant was that you said?
The 501Y.
501Y. 2 point, how many kilocalories?
Well, it's the differential between the normal ASIN and the Y is minus 2.2 kilocalories per mole. That's calculated, by the way. We've got we've just received. I told you we got the grant, eh? To measure it in real life.
Really?
Yes,
So this was the grant that you had to do the publication, the submission for within.
That's the award letter this afternoon.
Wow, South Africa works fast, man.
Well, they're moving very quickly. It's just the university still has to sign the contract. So the award letter has been done by the MRC, and now the contract is with our university officers. And so they've got to make sure that the university is going to get their pound of flesh and overheads and blah, blah, blah. That's all they're interested in.
Who published this thing about the 501Y?
It was, oh, golly gosh. I will tell, I can't look at now. I can't look at now. I will tell you. I will tell you. I will send you the slide. It wasn't Kurt, right? That was it was another guy. So Kurt did quite a lot of work, but he didn't do this particular bit of work. You know Kurt? Kurt
No.
Bidler.
Oh, right, yeah.
Did you meet him?
No, I know.
He works with Penny Moore at NICD.
Okay.
Very enthusiastic guy, by the way. You know what he's studying?
No.
He's studying synthetic antibodies against snake venoms.
Oh, right, that guy. Okay, I got you. Yeah, you know, I was just talking about that grant the other day. Because that's one of the NIH grants, right?
Yes.
Right. So, so it turns out, me and a colleague, we're working on a bill in Congress to so the United States government spends about $7 billion per year in foreign assistance to Africa. And so we're writing legislation that basically commits 1% of that to go towards science. And where 1% comes from, because the heads of states at the AU had committed to spending 1% of GDP on science about a decade or so ago. So the question became, so for the United States, that comes like to $70 million. So basically committing $70 million per year for science in Africa. And we've been talking about how does that get distributed? And there are some agencies like NSF that cannot make commitments to non-US institutions, but NIH can, right? So people at University of Free State and University of Cape Town have like normal NSF, normal NIH grants. So I rolled out the snake venom project as an example.
It's very interesting, and I mean it's going to be a lifesaver for many people. Do you know that a lot of people die due to snake poisoning? And
yep.
And
yeah.
the thing is that you know how the antivenoms are made?
Through what?
To the snake venoms.
No, how are they made?
Okay, there are 12 horses that are kept in halfway between Johannesburg and Pretoria on a farm, and they are bled as required, and their serum is processed and sent off to all the people that have ordered antivenom, and the serum lasts about a month. So you have to send it out to, I mean, it's a very special thing and very expensive and ludicrous process, because it's an unstable product, and it's it's a biological product, obviously, and it's not readily available. So with the synthetic antibodies, you'll be able to carry them in the field. So when a person gets, I mean, you'll be able to keep them in every village.
Right.
So when a person gets bitten by a snake, or a person is doing a hike or something, whatever, whenever the people get bitten, which is all over the place, they'll be able to pull the antivenom out of their bag and inject themselves.
Yeah.
And I mean, this is going to be just incredible for people who at the moment have to apply tourniquets and hope like hell that they can be airlifted to a hospital where they can receive appropriate treatment.
Right. Yeah, if I got bit by a snake, I'd be dead, because I'm sure my heart would be accelerating so much that, yeah, I'd be freaking out so much that I mean.
Well, the people do freak out.
The total exchange of my whole blood volume would make a circuit.
I mean, people here are scared.
Yeah. How come that guy didn't give a talk at this conference?
So only you know that.
I don't know that. That's it. I mean, it's an interesting project.
How come Kurt didn't give a talk? Because he considers himself to be super busy.
Yeah. Sure. Okay. Now he got that grant like in 2013 or 2014, right?
No, it was recently. It was just now, about 2 years ago, at the most, 2019.
Is it from? It's from NIAID. If
Yes.
or so you know how that stuff came about, right? It was when well, you were there when Hanekom came to town.
Yeah, he was at the ESRF meeting.
Yeah. Oh, well, maybe next year.
Hello. Yeah, what's it? Okay, 5 minutes to go.
Hey, Trevor.
Hello, who's that?
Zoe, how are you?
Hi, Zoe, how are you? Oh, you're looking great. And there's beautiful, beautiful crystals behind you.
Yeah, they're yours. Yeah, I quite like them. They look like snowflakes and they appeared around Christmas. So that worked out. Yeah, I was sorry to miss this morning session. It looked really good.
Well, it was very interesting, and there was a lot of people gave very good talks.
Yeah, I was bummed. I was stuck in a 3-hour workshop. I'd rather have been listening to structural biology presentations.
Anyway, it's quite interesting. It's quite interesting. There's a lot happening.
Super good.
And it would be nice to bring it all back to Africa.
Yes.
Yeah.
It's a diaspora, huh? Everybody's scattered all over.
You've got to put on your best Stellenbosch accent, eh?
Oh, it's been too many years. 21 years since I moved, you know? It's a long time.
Okay, so how many minutes? We've got 2 more minutes. I just got to get my program up so that I can actually keep track of events. Oh, there we are. A lot of Africans seem to end up in Sweden, hey?
Is it?
Yeah, it seems like it. We had a talk from Umia.
Oh, okay. Oh.
And, well, and it must be a friendly country. Although I can't imagine why anybody would go there because of the weather.
Yeah, the weather is special. You just have to trade off, I guess, quality of life with the weather. Although here in the south where we live, it is quite okay, actually. We do get a proper summer every other year. So.
Okay, so should we get started? How many people are signed in, by the way? Let's have a look.
15, including 16.
Yeah.
So
16 total.
Okay, well, I think we can start, and then people will join in as they come along. Okay, so the next talk is by Zoe Fisher. Welcome to the afternoon session. And what Zoe is going to tell us about is how to get started with a neutron crystallography and other neutron experiments. The interesting thing about neutrons that we've heard quite a lot about in South Africa in the recent past is that there is a move to create a second reactor in South Africa, or to rebuild the first reactor, and there have been several beamlines proposed, and there's also been quite a lot of interest expressed in different things that you can do with neutrons. So the idea is for Zoe to tell us more about it and tell us how to get started with work using neutrons. Okay, over to you, Zoe.
Yes, thank you. Let me. Wait, I can't share a screen. Host has to allow me.
Lawrence, you're the guy. Lawrence, you there?
Yeah. Okay, so this is good now.
Oh, okay. Thank you. Hey, there it is. Cool. All right, let's go for and laser. So yeah, thank you for the chance to speak to you guys today. It will be quite a very basic introduction to doing structural biology with neutrons, which is not such a familiar probe for many people, and I'm sure it isn't for you either. I am Zoe Fisher, and I am a scientist at the European Spallation Source, which is the next generation neutron scattering facility being built here in southern Sweden. So I'm a group leader for the deuteration and crystallization group at ESS, and I also hold an adjunct senior lectureship with Lund University. So my time is a bit split between the two. But my background is protein crystallography, first with X-rays, and then as a postdoc and beyond, I worked at neutron scattering facilities where I have continued research on an enzyme, carbonic anhydrase, to look at basic mechanistic questions around the enzyme mechanism, and also more and more looking at drug design using neutrons and X-rays in a joint mode. So that's a key used area for neutrons in protein crystallography. So today I will tell you a little bit about neutrons and how they are used for life science research, of course, with a very strong focus on structural biology. We'll talk a little bit about the properties of neutrons and why they're a good probe for hydrogen-rich materials, such as proteins, for example. I'll go a little bit into deuterium labeling and why we need to do that, and also why do we need big crystals, and actually how do we achieve those for neutron scattering experiments. And then I'll go really more into general examples of how protein crystallography has benefited from studies with neutrons. And finally, I have some slides on practical aspects, like what facilities exist, which kinds of beamlines are out there, how can you get access, and how sort of to get started. And hopefully there's some time for questions at the end. So a few years ago, to gauge the European landscape of neutron users, there was a massive survey done. And you can see the results of that here in this colored cartoon to show essentially the types of scientific disciplines where scientists are coming from when they use neutron facilities. And what you can see here is that life sciences is and was quite underrepresented in the vast field of neutron sciences. However, this is very rapidly changing, and in the last decade, the number of beamlines and structures that have been determined with neutron crystallography is growing extremely rapidly, with most results actually coming in the last decade. So there is a shift. We saw this with synchrotrons. Initially, they were very much focused on other types of science, and today, most synchrotrons in the world are heavily focused on MX, micromolecular crystallography. And I think we will see a similar shift in the coming decade as neutron sources get better, the beamlines become more numerous, and the measuring time is being reduced. But growth in this community has been always slow, and it always has been kind of a fringe technique. And there's a couple of things inherent to neutron facilities that really have manifested this result. The first thing is to recognize or to realize is that neutron sources inherently have extremely low flux compared to synchrotrons. We are orders and orders of magnitude fewer neutrons per centimeter squared per second than you can get out of a synchrotron or even a home X-ray source. Neutron sources are very weak when you have small samples composed of thousands of atoms. So this really puts a very strong requirement on the sample size. So you can compensate for the low flux by having massive crystals. And of course, this is not super easy to achieve. And it very much depends on the particular sample you are working with, your particular protein or enzyme or bit of DNA or whatever. Then this is compounded by the fact that there are relatively quite few neutron sources available for users globally. So throughput is really discourages people. If you have to wait a year or two even to get a sufficient number of beam days to do your experiment, it may be that you lose interest. You have many other experiments keeping you busy. And it takes, I would say, special dedication to plan and see a neutron experiment through, because you should be patient. And then while many facilities do have very good support laboratories to help users prepare their samples, they have very limited bandwidth as to how many people they can help. So that is also an issue, and we'll talk a bit more about what kind of support facilities are out there to help the life sciences community. So neutrons can be used in life sciences to actually look at a range of different length scales or sizes of molecules, if you will. Of course, the area shown here on the left is where I work. This is where my research interest lies. And this is to really look at the 3D atomic crystal structures of enzymes or enzyme or protein DNA complexes. And here we are interested in localizing hydrogen atoms in these structures to look at things like enzyme mechanisms, to look at what effect amino acid mutations may have. You can also use it to study drug binding, for example. And of course, here we're really talking about single crystal diffraction, and there are a number of beamlines that can support you for that type of measurement. Then we also have solution structures. So you can think about protein complexes, but in solution, not in a crystal, of course. These can be bigger systems, dynamic systems, flexible systems, things that are just not amenable necessarily to crystallization. Maybe you're also interested in the dynamics of a protein complex in solution. And here we're looking on the nanoscale. So it's a far lower resolution technique than looking at crystal structures. And yeah, you can see some very basic idea here of how you can use this. And there's a number of application areas where this may be useful. We can think about membrane proteins when they're in a micelle or detergent stabilized. You can think about mRNA vaccines when mRNA is stabilized also in a lipid vesicle, and so on. Then we can also do surface scattering methods using neutron reflectometry. And here the application for life sciences is very much focused on biological membranes or lipid bilayers. And you can reconstitute your favorite membrane protein into a lipid bilayer and study it with reflectometry. And here you can add membrane disrupting agents, membrane penetrating peptides, such as antimicrobial peptides, and study the effect they have on the membrane using this method. Today, I will mostly talk about the crystallography stuff. And the beauty of neutrons is that they can be produced in these facilities with a range of energies or different wavelengths, which really allows you to probe different length and time scales of the particular sample you may be interested in. And this slide is a bit messy, but it's intended to demonstrate the resolution and the size of the thing you can study, all the way from individual atoms in crystallography, all the way to intact living things, such as this beautiful neutron image of a plant, a flower, and everything in between. And what is shown here is how these length and time regimes overlap with complementary methods that researchers are routinely and more frequently using in their labs. Anything from X-ray diffraction to small angle X-ray scattering, dynamic light scattering, to microscopy, both optical microscopy, cryo-electron microscopy, and so on. And we can essentially produce neutrons in all of these flavors to match what you are able to achieve with other techniques. And it gives a very complementary view. And the reason neutrons are so complementary is that they have this unique behavior compared to other probes, such as light or X-rays, in that the scattering of a neutron off a particular element does not depend on the number of electrons that atom has. It actually sees the atomic nucleus. So the magnitude of the scattering is completely independent of the atomic Z number, which is very interesting when we look at the light atoms. So here is a very reductionist view of the periodic table as seen by a biologist maybe, where you can see with X-rays, you have this very predictable increase in scattering power depending on how many electrons you have as you walk your way across the periodic table. Neutrons, however, see the atomic nuclei, so they're completely blind to the number of electrons, and every atomic nucleus scatters the neutron to a different extent, but all actually to a very similar extent. So here, for example, you can get equivalent scattering from a carbon and an oxygen as you can from deuterium, the isotope of hydrogen. Because neutrons see the atomic nucleus, they are very sensitive to different isotopes of the same element. And again, for us as enzymologists or protein crystallographers, we are very much interested in hydrogens. And you can see that X-rays are essentially a useless probe if you are interested in localizing specific hydrogen atoms in your favorite protein crystal structure. Whereas neutrons have a very strong scattering. They're very sensitive to hydrogen and the isotope deuterium. One bad thing about hydrogen that is not illustrated here, but you can actually see the numbers, is that it has a negative coherent scattering length. And if we replace it with the isotope deuterium through a process called deuteration, then you can get very strong positive scattering actually, but not really introduce a significant chemical change to your particular material you're studying. So for this reason, it is incredibly not only beneficial, but often essential to deuterate your sample for neutron experiments. And it is really, really good if you can replace as many of those natively abundant hydrogens with the isotope deuterium. And we can do this in different ways. And I'll tell you a little bit more about the methods we use in my lab. So the purpose of deuteration very much depends on the technique you are going to use. Excuse me. On the left here, we can see that for protein crystallography, we exchange the hydrogens for deuterium to really profit from this superior strong positive scattering we get from the deuterium with neutrons. And if we do that in a systematic and careful way, we are all of a sudden able to see very easily, even at sort of mediocre resolution, where many of the hydrogens are. So instead of just a blob of spherical density for the oxygen, only of a water molecule, as you would see with X-rays, you can now actually start to see features that indicate where the hydrogen, or deuteriums in this case, are sitting on these solvent molecules in a protein or enzyme active site. Here is an extreme example, which I like to show, but it is quite unrealistic. This is an ultra high resolution neutron crystal structure of crambin, something like 46 amino acids. It's barely what I would call a protein. It's probably a large peptide. But here we managed to get extreme resolution, and you can really see these amazingly well-defined water molecules, which then allow you to map very precisely and unambiguously the hydrogen bonding interactions between these solvent networks. So this is always what we strive for, but this kind of resolution is not achievable with most ordinary protein systems. Then we're also able to distinguish, for example, between neutral and charged amino acid residues. And here we can see lysine side chain and how the map actually looks when it is fully charged. And this is something that could be very useful for enzymology type studies. For the other methods I mentioned, small angle neutron scattering, or SANS, or NR, neutron reflectometry, the purpose of deuteration is not to see the actual hydrogens. The technique is far too low resolution for that, of course, but we instead use deuteration as a tool to allow us to enable contrast variation. And here you may have a complex of two different components. You selectively label one with deuterium and you keep the other one as the fully ordinary proteated variant. And you can then dissolve these in different concentrations of H2O and D2O, and effectively at a specific amount, you can actually mask out one of the components depending on its deuteration status. So here you can see the scattering length density for a particular material and at which stage it becomes invisible when you vary the ratio of H2O to D2O. So here, when you cross the black line here, in 60% D2O, for example, you can make DNA completely invisible in these kinds of studies. And then you can study the shape and behavior of the thing you're interested in, but in context of its real native binding partner. You can think of protein complexes, for example, or transcription factors bound to bits of DNA. And this is illustrated down here. So just by varying the concentration of deuterium in your measurement, you can then mask out one or the other material. So still, you need deuteration, but the purpose is quite different. There are many labs that help people and offer deuteration as a service to their users. There is I would say, an informal network called the Deuteration Network. It was started by the ESS. We had an EU grant to essentially coordinate the deuteration facilities located at different neutron scattering facilities into an entity to try and get a view of what everybody is doing, what everybody is offering, but also really to try and reduce redundancy, that we don't all offer and focus on the same thing, so that we each sort of have a niche area or something we're good at, and that we can offer to users without duplicating the investment by the various governments, of course, into offering these services. So the original DUNET partners involved ICES in the UK, the ILL in France, ESS as the organizing node here in Sweden, and Jülich in Germany. Since the early days, which is now some 6 years ago, the network has massively expanded. We added JPark in Japan and ANSTO in Australia as observers. They both run also deuteration labs, but they weren't part of the original European grant, of course. And since then, we have also added deuteration labs in Korea and a couple of other places, I think. So the network is really blowing up. And the information on our website, deuteration.net, you can check out information about each of the deuteration facilities. So the website is a bit more up to date than the image shown here. And we also run a Twitter account where we may tweet about random deuteration news. So I invite you to check those out if you are interested. Shown in red is sort of the various areas where each of these support facilities tend to specialize. So for example, the one in ICES in the UK, they really only focus on chemical synthesis of small organic deuterated molecules, whereas the ILL is very well known internationally for their biological deuteration laboratory and so on. ESS does a little bit of everything. We have a chemical deuteration lab, we also do biological deuteration, and we offer support for crystallization as well. This is my group. So here you can see the different kinds of support we actually offer to the user community. So we have 3 areas, like I said. We have 3 core staff members. There's myself and my colleagues, Hannah and Anna. And then we have 2 postdocs that are funded by grants, but they're doing their work in our lab on deuterated materials. Janie is focused on melittin compound in bee venom to look at its anti-cancer activities. And Jai Fei is a synthetic chemist, and he's looking at surfactants, photo-switchable surfactants that, of course, are deuterated. And Fatima is just on a short-term contract as an extra set of hands in the lab for us. So we do chemical deuteration, where we can synthesize small organic molecules, a number of different things we've made, small monomers, pyruvate, lactic acid, and others, all the way to oleic acid, POPC, POPE, phospholipids, and so on. We also can extract and separate phospholipids from living systems such as yeast to provide more natural lipids to the people doing SANS and reflectometry, as opposed to the synthetic analogs you can purchase commercially. Then in our biolab, we grow all the cells. So here we culture algae, bacteria, ordinary E. coli, and yeast, specifically Pichia pastoris, under deuterated conditions, depending on what the user needs. We can prepare deuterated plasmid DNA for example, SANS experiments, and we can also prepare deuterated proteins for crystallization or for reflectometry or SANS or whatever. Yeah, and then for crystallization, we share our crystallization laboratory with a group at Lund University. They are really more focused on teeny tiny 200 nanoliter drop high throughput screening methods for synchrotron applications, whereas we do more low throughput and careful scaling up and optimization for just growing huge crystals in my lab. But of course, the same underlying methodology exists. So we do share a lot of equipment, microscopes, incubators. We have access to plate hotel to image plates automatically, as well as some liquid handling robotic systems such as the Mosquito and the Oryx 8. So we're a small team, but I think we can offer quite a lot. So for biodeuteration, like I said, we grow different types of organisms. This activity is housed in the biology department at Lund University. So it's very easy and nice. We have access to tons of equipment. For proteins and DNA, we mostly do this in E. coli. It is the most common and cost-effective way to deuterate proteins and DNA. Then we can also do yeast. We typically grow these in several batches to order as a user requires material deuterated from yeast. And the main projects we are dealing with at the moment is to optimize the methods for extracting natural deuterated lipids from the yeast cell paste. We also have a project coming up later this year where we would like to look at cardiolipin, and their bacteria is a better source for that deuterated lipid. And then, of course, we really grow the algae for the total cell extract that we can prepare from it. And we use that algal cell extract to prepare rich media, which is a little bit more like Luria broth for growing E. coli, as opposed to minimal media under deuterated conditions, which is quite stressful to the cells. And we really see a 50% drop in the yield of the protein and stuff we can get out of the cells. So we basically have a renewable way, just using LEDs and minimal media to grow algae on a near continuous basis. And then we hydrolyze them and use that for making this rich media. So biodeuteration very much depends on the application and technique, how much deuterium content you need in your sample. I will say it is quite expensive to prepare. Heavy water is very expensive. Here we pay something like €350 for 1 liter. And you can imagine when you're preparing 6 or 9 liters of cell culture media to do a preparation to purify proteins, it gets extremely expensive. So we're always looking for ways to minimize the cost, but really maximize the amount of deuterium in the sample, depending on what the user needs. The shown in red is by far the cheapest and easiest way that consumes the least amount of D2O. And here we are essentially doing HD exchange. So here you can exchange many of the labile hydrogens in your system with deuterium by simply dissolving or diluting your protein solution, for example, in deuterium-containing buffers. As the protein is already folded when you do this, you get between 20 and 30% of the labile hydrogens may exchange. Usually this is good enough. Many enzyme active sites, for example, are super solvent exposed. And so you get very good exchange in the areas of interest and typically very poor exchange, like in the buried backbone regions where you have tight hydrogen bonded networks that are not very solvent accessible. And of course, your aliphatic hydrogens here will not be replaced. So hydrophobic residues such as phenylalanines, valines, isoleucine, and so on will stay as just CH. Then we can do different levels of partial or full deuteration. That costs a little bit more, but is quite possible. For partial deuteration, number 2 is especially beneficial actually for all neutron methods. This is now becoming common in SANS and refractometry as well, where you use D2O to prepare your medium, but instead of using super expensive deuterium labeled glycerol or glucose as your carbon source, you actually use unlabeled carbon source. And the effect is that you get 55 to 80% deuterium incorporation into the final protein. And this is quite okay. We do this in my lab frequently, again, depending on what the material will be used for. And we also have a rotary evaporator system. So we recycle and reuse our D2O many times to also try and reduce the cost. Then in number 3 and 4 here, we get to the really expensive parts. And this is also where the cells start suffering. They start to not grow very well, they grow very slowly, and the yield of the material we try to extract also tends to reduce dramatically. And here we are going really for 99.9% deuterium deuterated conditions, including using labeled carbon source. So we purchase per deuterated glucose or glycerol to feed the E. coli or yeast. And another way to do this that is slightly cheaper and more sustainable is to just grow the algae in heavy water that we can recycle many, many times, and we don't have to feed them anything. They take CO2 from the air, and we just have to shine an LED on them for roughly 12 hours a day, and they grow like crazy. And they're quite tolerant to 99% D2O. And then we can sacrifice them by preparing essentially a soup and feeding that back to the E. coli instead of purchasing this expensive labeled glucose. And we do all 4 of these in my lab, again, depending on discussion with the user, what it is they want to use the material for. So in addition to this constraint of we need to deuterate our samples to whatever extent necessary, for crystallography, we also need to grow big crystals. And big crystals means big compared to what is normal or routine for X-ray applications. So typically today, I'm sure many of you do experiments at the ESRF and similar. You're fishing crystals that are 10 to 100 micron, probably on edge. In tiny loops, you plunge them in nitrogen and you get beautiful data. For neutrons, we have to be 1 to 2 orders of magnitude bigger in volume. And that is seriously not easy. So on average today, most neutron instruments really need your sample to be 0.5 millimeter cubed in volume. So you can calculate how to get there. That is quite a big crystal. You usually don't need a microscope to see them. And the volume calculation, of course, depends on the actual shape of your crystal. So something shown here in the top is myoglobin. These are incredibly chunky. So you have roughly the same dimensions in all 3 directions. So you get a very nice big volume. Here is an ordinary ruler. So this thing is a couple of millimeters in each direction. So you get 6, 7, 8, 9 cubic millimeters in volume. But when you get to some of these systems that I'm used to working with, where you get plates, yeah, okay, it is 4, 5 millimeters long and maybe 1.5 wide, but it is very thin in the 3rd dimension. So when you calculate the volume, of course, you end up with a crystal that is 0.1 millimeters cubed, even though it looks massive when you're fishing them under the microscope or without the microscope, if everything goes well. So that is a challenge. So on average, you want 0.5 to 1 millimeter cubed. Bigger is always better, but that is sort of the lower limit. In exceptional cases, if you have very well-ordered crystals with very low solvent content, you can get away with something like 0.1 millimeters cubed, but that's quite rare. And a good indicator of this is how well your crystal diffracts X-rays. So if you have something where you have low solvent content, something that diffracts X-rays like crazy to atomic resolution, then you have a pretty good chance about having success with neutrons in this range, but not by any means guaranteed. So to get to these sizes, we use a bunch of different strategies. Of course, we usually start with standard hanging drop crystallization, but here you're really limited. I think the max volume you can aim for here is 10 to 15 microliters. Maybe if you have a lot of PEG, for example, and it's super viscous, then you may get away with that size. So this is really good for screening for us, and we always aim for 10 microliter drops to just see that the condition works in that kind of volume. And then we immediately have to transition to some kind of sitting drop method. There you have, the sky's the limit as to how much protein solution you can add in a sitting drop. And we start with 10 to 30 microliters, and then you only go up from there, depending on the kind of hardware you have available that can hold a drop of a certain size. And this is very successful in our hands. We have the most success growing big crystals with vapor diffusion, but with massive sitting drops. And they can be up to 1,000 microliters in volume to get these kind of crystals shown up here. Of course, if you have batch technique, that is really good. Batch is very easy to manipulate. And in theory and in practice, there's no limit to the volume you can do. So here you have no evaporation. You cover your drop with paraffin oil, but it means you need a really good handle and understanding on what your precipitant concentration should be when you start. And dialysis as well. You can buy dialysis buttons from Hampton Research, for example, that have a range of different volumes. I think you can buy up to 350 microliters. So you can prepare your crystallization drop, pick up the button when it's prepared with tweezers and put it in different beakers containing different precipitants and different concentrations of precipitants. And they are transparent, so you can actually look through them to see if you're getting nucleation and crystal growth. This one is a bit fussier, and I can tell you it's very difficult to harvest crystals from this setup. So that's a bit tricky. So really, I would say batch and sitting drop is probably the smoothest way to prepare big crystals for neutron crystallography. So we usually start in this kind of size regime, and then we systematically increase the volume and constantly monitor to make sure that the crystals appear. When you get to big volumes, the kinetics of the system massively slow down, so it takes a long time to get nucleation and crystal growth. So you should also hopefully be working with a protein that is quite stable. So neutron protein crystallography is used to determine the atomic crystal structure of protein molecules. It is single crystal Bragg diffraction, just like you would do with X-rays. The difference here is depending on the source you go to, you can actually do monochromatic, so single wavelength MPX, or you have access to a range of wavelengths in the Laue regime. So here we call a limited wavelength range, let's say 2 to 4 angstroms as a range that you shine on the crystal, quasi-Laue or a pink beam, or true Laue where you shine a white beam or the full spectrum on the crystal. And my old beamline in the US, we could do 0.7 to 7 angstroms simultaneously on the crystal. And that's quite useful because you get a lot of reflections at once, thanks to all the wavelengths. So it can reduce the number of images you need to collect to get a complete structure. So there are some nuances depending on what beamline you visit, but by the time you end up with a processed file containing your structure factors, everything works the same way as it does for crystallography. Structure refinement and modeling, you use all the same software tools, you use COOT, you use Phoenix, and you do it in a very similar way as you do for X-ray crystal structures. With the exception, of course, that you do have now 3 atoms for every water molecule as opposed to only an oxygen. So you almost double the amount of atoms in your protein databank file to account for all these additional hydrogens that now actually are not silent in your structure. They're actually contributing massively to the scattering of your crystal. But it is important to remember the samples have to be big. This takes some effort and care. And the data you collect in the industry is quite weak. Remember, I talked about the inherent flux of these facilities being quite low, which means your signal to noise in the measurements is quite low. Your statistics are not quite as nice when you merge your crystallographic data. And of course, the data collection takes forever. So with Trevor at the moment, we are collecting data in Grenoble on one of their Amadeus crystals. And I think so far that crystal has spent almost 20 days on the instrument. And the data set currently is not that complete. So we still need probably another 7 days. All in all, I think it will probably be 30 days of beam time to get a reasonably complete data set to 2 or 2.2 angstrom. So that's what we're dealing with. Even though Trevor and Stan's Amadeus crystals diffract X-rays like crazy to almost atomic resolution. With neutrons, the story is a bit more complicated. And it really is all driven by the flux problem. However, if you overcome these sample preparation issues, neutrons are incredibly useful and complementary to everything else you are probably already doing. They give us the ability to directly visualize hydrogen atoms and all their interactions, such as hydrogen bonds. Once you can see hydrogen bonds, you can actually really map them in water networks and see how solvent is connected to the protein. The presence or absence of hydrogen atoms on amino acid side chains, for example, also tell you the electrostatics and the charge state of the surface of your protein or the active site of your enzyme. And you can do this in a very precise way. We can also very precisely map the details of interactions between ligands, inhibitors, substrates, products, and an enzyme, for example. Again, because we have this ability to see indirectly electrostatics, but also the ability to directly see the hydrogen bonds involved. And as we know, many enzymes use water as a catalytic helper, and neutrons give us the ability to very easily distinguish between water, hydronium, hydroxide, or just a naked proton. And this is because the scattering from each of the different numbers of hydrogens in these entities is very, very different. And in practice and reality, you will never see a proton, of course, with X-rays. And with X-rays, there's no way really to distinguish between a hydronium, a hydroxide, or an ordinary water molecule, whereas neutrons can do that. So for structure function studies of proteins, it is incredibly important to know where the hydrogens are. 50% of the atoms in our proteins are in fact hydrogens. They are integral parts of the structure, but also the function of enzymes, for example, and they are responsible for making something positive, negative, or neutral. And the transfer of hydrogen is also a key part in many types of enzyme catalyzed reactions. Yet, using most of the techniques we use ordinarily, we just don't know where they are. And that's a shame. So here are just, again, a couple of pictures to show you how this looks in practice. Again, on the left here, this is the highly idealized situation of Cranbourne, but you can see this was something like 0.8 angstrom resolution X-ray structure from APS in the US. And even at this resolution, we still see mostly spherical blobs for these water molecules. So there's very little hope that you can map hydrogen atom positions using X-rays alone. But neutrons, of course, readily reveal where those atoms are. These are the same set of water molecules. Here is an example of the first time a hydronium ion was observed in an enzyme published in Angewandte in 2011. And like I said, you can use this to distinguish between these different things. And then finally, for protonation state, you can see down here, here is a histidine side chain, for example, that has both deuteriums on the imidazole present, indicating that it is indeed charged. And same for lysine and so on. So that's a very powerful use of neutrons if you have questions in your particular system that relate to these kind of issues. For ligand binding, it also is very interesting. It is mainly interactions involving hydrogen atoms that actually govern ligand and drug binding to target molecules through a number of mechanisms, desolvation, hydrogen bonding, and so on. In addition to this, 25% of marketed drugs today exist in more than one tautomeric form. And they can also be protonated or deprotonated when bound to the clinical target. So for example, if you co-crystallize something with a small molecule drug. So there's a lot of nuances that we simply can't see if we are only looking at X-ray crystal structures. And here is some work we did ages ago on acetazolamide, and we know in solution it can occupy multiple charged states, but we don't know which form is actually bound in the numerous crystal structures of this thing bound to carbonic anhydrase in the PDB. So knowing which tautomeric form and what is the charged state of a ligand or inhibitor when it's bound is very important for medicinal chemistry. If we're going to improve a drug or maybe even synthesize something from the beginning. Having a handle on the electrostatics we're dealing with is quite important. So again, we can use neutrons then to look at these specific binding interactions because we can see all these variety of features related to protonation and charge. Here is a beautiful arginine that is fully exchanged. So you can see strong scattering from all these deuterium atoms on the side chain. We are able to see the hydrogen bonds it makes, not only back to the protein itself, but also to the ligand sugar shown here. Same, there's been a lot of work done on HIV protease, where they've looked at amprenavir bound and also mapping exactly the deuterium atoms involved with the 2 catalytic aspartate residues. And neutrons have actually contributed a lot to the knowledge of HIV protease inhibitor design. So on to the practical stuff. What kinds of instruments exist and how can you access these facilities? So today for MPX, it's quite limited. The last time I checked, I think there were something like 180 crystal structures deposited in the PDB, determined by neutrons, and that is all structures ever determined by neutrons going back to the 1960s. But I will say more than 50% of those have been done in the last 5 years. So it is accelerating, beamlines are improving, detector technologies are improving, and the throughput is getting much better. But there are still only a few instruments in the world. There are 4 beamlines located at research reactors. There's the 2 very, very good ones in Grenoble, Ladi, and the new one, DALI. DALI was just being installed and commissioned at the beginning of the pandemic, and they had to pause the installation and commissioning work for that beamline. I'm not sure if they've started yet, but it will essentially be a clone of Ladi. So ILL will soon double its capacity for collecting these kinds of data sets. There's the monochromatic beamline BioDiff in Munich, and then there's Imagine, which is very similar to the Ladi instrument, but based at Oak Ridge, Tennessee in the United States. Then today there are 2 instruments built at spallation neutron sources. That's the kind of neutron source that the ESS will be. There's IBIX in Japan, shown down here, and MANDI in the US, shown here. And of course, we are building NMX here at ESS, and it should be ready in a couple of years. And the instrument shown up here was the PCS in Los Alamos, New Mexico, and that's actually where I worked and was a beamline scientist for something like 7 years before we moved to Sweden. That instrument has been retired, and this detector is actually now at Oak Ridge being used, I think, for powder diffraction beamline. Yeah. So those are your options for instruments. They look very different. They are very different, but the data you get out is essentially indistinguishable between these beamlines. So you should just aim to go where you can actually get beam time. That is the biggest rate limiting factor, I would say. Almost all these facilities, or all the ones I talked about on the previous slide, operate on a peer review proposal-based system. Some have restrictions on the national balance of the proposal team, or they may exclude specific countries that are not members to the funding of the operations of these neutron scattering facilities. And the proposal and the composition of your research team on a proposal vary by facility, but it's best to check with each one. They are quite clear about this upfront on their websites, so it's not difficult to figure out what you need. Usually facilities issue 1 to 2 calls for proposals a year with a hard deadline, and it's good to pay attention to these. But a handful also offer rapid access if you need a short amount of beam time and it is very urgent. Like you are about to make a Nobel Prize worthy discovery, but you just need 2 days of neutron beam time. Or you have something new that has never been done before, or your sample is extremely sensitive and you can't keep it until the next beam time comes around. So these are the kinds of cases where you may qualify for rapid access. And these are typically open all year round. So you can apply any time during the year. Usually it is just for a limited amount of beam time. Here I show a screenshot, for example, how the Apply for Beam Time website looks for the ILL. And it is very complicated because the ILL structure and funding is quite complicated. So here you can see that they have many different types of proposals and groups that can apply. And they say who is eligible for this particular type and how many deadlines a year they have. But I invite you to click around. Proposals usually are 2 to 3 pages, including figures and references. Again, facilities have quite strict rules on how long it should be. You really should explain the current state of the knowledge and clearly motivate why you need neutrons and what scientific question you have that cannot be answered with any other technique. And the proposal should clearly explain what kind of measurement you're interested in. So very specifically, what samples you have, how many days of beam time you need, and why. So it should be quite well motivated. And finally, if you're lucky, if your proposal is reviewed and accepted by the facility, you will be notified when your beam time may be. And there are quite some logistics involved, so plan in advance is highly recommended. There's things like, well, there used to be travel. I think these days most is mail in, but you have to arrange your travel, your accommodations. You may need to do additional training for lab access, how to ship your sample, and if you need special equipment for your measurement and so on. And the local contact or the instrument scientist and the user office are your best friends. They will really help you during all the steps for planning the experiment until you actually arrive. And for data analysis and interpretation, I really encourage you to engage an expert or get a collaborator that knows what to do with these kinds of data to help you out. So finally, that's all I had. So I would like to say thanks for the invitation and thanks for your attention. And I know I'm a bit over time, but maybe there's 1 or 2 questions.
Thank you very much, Zoe. I must say that my own experience of working with you has been spectacular. You really, in the session that we had the other day, you opened my eyes to, I was a complete neutron skeptic until the other day. And what you showed me that was possible to see in the active site of our enzyme was a complete revelation. So I'm looking forward to getting the complete data set. It's about 65% or 46% in the outer shell.
Yes, we should get there.
It is completely remarkable. And thank you very much for all of that. Does anybody have any questions for Zoe? I don't see any hands up at the moment. Are there hands up? Okay, then that being no hands up, I'll thank you very much.
Yes.
For
Thank you. And I'm sticking around for the next time, I mean, for the rest of this today's session. So if there are questions later, we can chat at the end.
Okay, thank you. Thank you very much then.
Thank you.
Okay, so then next up is John Rubenstein. Have you, Lawrence, have you made John able to share his screen?
Yep, I can do that.
Okay, so John is from Toronto in Canada, and he's an electron microscopist who has made huge contributions to the field. And in particular, the reason that he's especially interesting to South Africans is because of his work on TB-associated proteins and the amazing strides that he's made in understanding ATP synthase, which you will see in a moment is the most incredible molecule that's ever been determined, I reckon. So over to you, John. I think that we're looking forward to this.
Thanks, Trevor. Can you see my screen okay? Can you see my presentation, Trevor? Or anybody?
Yes.
Yes, okay. Fantastic, thank you. So I'm really honored to be here. I wish it could be in person so I could meet a few more of you in person, but happy to make do in the situation to tell you about my lab's work on mycobacterial ATP synthase. So as Trevor mentioned, both he and I have worked for many years in this field of electron cryomicroscopy, really trying to develop the methodology to the point where we get some high resolution insight into biological molecules. And we have basically over the past few years achieved that goal. So in today's lecture, I want to tell you primarily about the sorts of structures that we obtain rather than dwell on the physics or the computation that went into that. The process for us starts with this 200 kilovolt electron microscope. And this is identical to the microscope that you have in Cape Town. In fact, the one that you have in South Africa is actually the one that I used as a graduate student in the UK when I was doing my PhD. I understand it has some issues now, but it's really, for us, has been a workhorse in Toronto for screening our specimens. Our pipeline is maintained by a former postdoc, Dr. Smear Bendelekber, who make sure our microscopes are in good working order. And we use this rather old microscope to screen specimens and get them into a condition where we can take them to a state-of-the-art modern microscope to get high-resolution insight. Not the same sort of high resolution that we saw in Dr. Fisher's talk a moment ago with beautiful sub-1 angstrom resolution, but still high enough resolution in order to be able to build atomic models for macromolecular assemblies. So once we've prepared a specimen and screened it with this now rather old microscope, we take it to a shared microscope facility. So this is a Titan Krios microscope, a 300,000 volt microscope with a special direct detector device and really highly automated. And there we can collect a high resolution data set. This is one microscope that's a shared resource for the Toronto community and the Ontario community. It really only sees the best specimens that have been pre-screened. There are 3 of these microscopes in Canada. One might argue that's a little bit too few for the size of the country. There are probably over 100 in the United States, and one might argue that that is probably too many for the size of the country, especially if they're limited to looking at the best and pre-screened specimens. But with a shared resource like this, you can obtain a data set that allows you to calculate a high resolution map of a protein. So this is about 3.4 angstrom resolution, and that's sufficient resolution to build an atomic model for that protein. And nowadays we can do that really in less than 2 or 3 weeks. So in our best cases, if we really understand the sample we're working with, we can go from a protein purification to a high resolution structure in a matter of days. So what I'm going to tell you about the way that we use this pipeline is the biological direction that my lab has worked on for quite a few years. The overarching theme of our research has to do with large protein complexes that exist at energized membranes. We can divide that further into vacuolar type ATPases, which will not be the focus of my talk, but I'll give you a little overview of some of our recent structures, and then really focus on the ATP synthases and what we've looked at for the eukaryotic enzymes, the bacterial enzymes, the mycobacterial enzyme, and most recently, the interaction of the mycobacterial enzyme with the TB drug bedaquiline. So in the 1970s, Peter Mitchell proposed that in biology, energy could be stored as a transmembrane proton gradient. This will have an electrical component because protons are charged, so an inequilibrium across a membrane produces an electric field, and also a chemical component because of this ion gradient. And when Mitchell proposed this, it was really a controversial idea, but when it became fully accepted, it changed biology fundamentally because suddenly cells were not bags of enzymes. They could be compartmentalized with membranes maintaining electrochemical gradients across them. The context in which Mitchell was thinking about his chemiosmotic hypothesis was that of respiration. In cellular respiration, all of the food that we consume, or almost all the food that we consume, is catabolized, is oxidized in order to produce this chemical NADH. And NADH serves as an electron source for the electron transport chain. So the electrons from NADH are passed sequentially from mitochondrial complex 1, that's found in the inner membranes of mitochondria, to a small molecule quinone, reducing it to quinol. The enzyme complex 2 also oxidizes succinate to fumarate and uses those electrons to reduce quinone to quinol, contributing to the pool of reduced quinol in the mitochondrial inner membrane. And then the electrons from quinol are taken to complex 3, passed via cytochrome C to complex 4, and ultimately used to reduce oxygen to water. So this is the reason why we need oxygen in biology. It's as a final electron acceptor for this mitochondrial electron transport chain. During the passage of electrons, 3 of the complexes from the inner mitochondrial membrane pump protons across the membrane. So complexes 1, 3, and 4 will couple transfer electrons to transmembrane proton pumping, and that builds up this positive outside the mitochondria, negative inside the mitochondrion electrochemical potential. The ATP synthase allows protons to go back through the mitochondrial membrane and uses the free energy liberated from the translocation of protons to make ATP, the chemical energy currency, from ADP and phosphate. Another enzyme complex known as the vacuolar type ATPase carries out the same reaction as the electron transport chain. It pumps protons across a membrane. Here, it uses the energy liberated from the hydrolysis of ATP to ADP and phosphate to pump protons. And the way that it does that is using a mechanism that's extremely similar to the ATP synthase. It's really a lot like the ATP synthase, except running in reverse. So it's not making ATP, it's hydrolyzing ATP and pumping protons rather than having proton translocation drive ATP synthesis. And so I won't dwell on this complex, but really I can't not talk about it because what we learn about this enzyme really informs our work on the ATP synthase and vice versa. And so the lab's research really has the synergy looking at all of these enzymes together. And so we've solved a number of complexes over the past 5 or 6 years, starting with the yeast VHPase, the work of a graduate student, Jinhua Zhao, and Samir when he was a postdoc. And here, even though we weren't getting to the resolutions where we could build atomic models, Jinhua was able to do something quite remarkable and separate out computationally the different conformations of the enzyme that exist simultaneously in our specimens and make a movie of what the enzyme looks like as it goes through its rotary catalytic mechanism. So here, ATP hydrolysis in the catalytic region drives this rotation of this rotor subcomplex, and it's the rotation of this rotor subcomplex that drives proton translocation across a membrane, which would be found over here when this is incorporated into a membrane. Mohammed Masab Jafari, a former postdoc, now faculty at the University of Toronto, was able to isolate just the membrane region of the complex, and because it was more structurally homogeneous, he was able to get that to a resolution where he could build a full atomic model. And then Thamia Vasanthakumar has improved the resolution of the intact structure and looked at how lipid binding occurs and shown that as the enzyme rotates, we actually expect to see binding of sterols to this ring and then the sterols being kicked off as the ring rotates. Most recently, just last year, Yazan Abbas, a postdoc in the lab, used a method that we had developed to isolate the enzyme from mammalian brain. And this is where the enzyme is used to load synaptic vesicles with neurotransmitters. And again, we could observe all the different rotary states of the enzyme, now getting to the resolutions where you could build full atomic models in the intact complex, something that we couldn't do 5 years ago looking at the yeast enzyme. So that's the vacuolar type ATPase, but what I really want to focus on for my lecture today is the ATP synthase. And just like the vacuolar type ATPase, the ATP synthase is a rotary enzyme. So it can work in either mode of proton pumping or ATP synthesis. In the mode of proton pumping, you would have ATP hydrolysis, occurring in this soluble catalytic region. That ATP hydrolysis causes conformational changes that drives the rotation of this rotor complex, and it's the rotation of this rotor complex against this green stationary stator complex that causes proton translocation across a membrane. In the mode of ATP synthesis, it's the translocation of protons driven by the proton motive force that induces the opposite sense of rotation of this rotor subcomplex that drives conformational changes in the catalytic region, and that leads to the synthesis of ATP from ADP and phosphate. And so a graduate student in the group, Anna Zhou, was the first to look at the mitochondrial ATP synthase since getting the improved detectors that let us get to the resolutions where we could see secondary structure. This was a collaboration with my former PhD advisor, John Walker in Cambridge, one of my former PhD advisors. And we were able to collect a data set where we could see the secondary structure of this, of really all of the proteins that make up the ATP synthase. So what we're looking at here is a cross section through the catalytic region, and you see these two white dots. Each dot corresponds to a single alpha helix penetrating between the catalytic subunits of this catalytic region. And we were able to separate out these different rotational states. So you could see that these dots are in different conformations, and we could play through those different rotational states as a movie, and you can see that central rotor rotating within the catalytic region and driving the conformational changes at these catalytic subunits that lead to the synthesis of ATP from ADP and phosphate. The mitochondrial ATP synthase, it turns out, does not function as a monomeric complex. So there are many subunits that make up the complex, but it doesn't even stop at that point in the mitochondrial membrane. The ATP synthases actually assemble into large dimer ribbons. So this is a classical histology image of a mitochondria. It's actually rather misleading. If you look at a 3D reconstruction, some work from Werner Kuhlbrandt's lab at the Max Planck Institute in Frankfurt, you can actually see that the mitochondria are, when you look at them in 3D, the cristae of the mitochondria form these rather complex structures, and the ATP synthase particles actually assemble as ribbons of dimers. This was actually known from 2D images from Richard Allen's group at Vanderbilt University from the 1980s, where if you do freeze fracture electron microscopy, you can see these ribbons of dimers. And Allen was way ahead of his time and proposed that the cristae form these complicated tubular structures with the dimer ribbons actually bending the structure. And we know from studies in yeast that if you knock out the subunits responsible for dimerization, mitochondria actually lose their cristae and become onion-like structures. And this dimer structure is true for mitochondria from any organism that has where the studies have been done. So Hui Guo in my group was able to isolate at the time just the mitochondrial ATP synthase dimeric membrane region. By getting rid of the catalytic region, he got rid of the conformational states that are interesting and informative, but tend to blur out the structures. And by doing that, he was able to build this first atomic model for the membrane region of the ATP synthase, of any ATP synthase. And that was actually extraordinarily informative because John Walker's group in the 1990s had solved the crystal structure of the catalytic region, and he had won a share of the Nobel Prize for that work. And so we could take a low resolution structure of the intact mitochondrial ATP synthase dimer and Hui's high resolution atomic model of the membrane region and just fit them together, and they fit perfectly. And that gave Hui this first atomic model for the intact mitochondrial ATP synthase dimer. That allowed us to look at the mechanism that couples proton translocation driven by the proton motive force to the synthesis of ATP from ADP and phosphate. And so the model that existed before the structures, but which is borne out by the structures, is that there are two offset half channels for protons to cross the lipid bilayer in the membrane region of the ATP synthase. There's one half channel over here and an offset half channel over here. And during ATP synthesis, protons go halfway across the lipid bilayer and bind to conserved carboxylic acid residues, glutamates or aspartates, that can exist either in deep protonated negatively charged forms or protonated neutral forms. So the proton will bind to a deep protonated glutamate, neutralizing the charge, and that neutralization of the charge of the glutamate allows that glutamate to enter the hydrophobic environment of the lipid bilayer. Of course, a negatively charged residue is not very happy entering a hydrophobic lipid bilayer, but if you protonate it, it's still highly polar, but it's more thermodynamically favorable that it can enter the lipid bilayer. So protons will go up through this half channel, neutralize the glutamate, allowing this ring to turn, and the rotation of this ring brings protonated glutamates to a second offset half channel where something happens, the pKa drops, and the proton is released to the other side of the lipid bilayer. And the structures that Hui determined really bear out this model, where we can see clear paths through the structure of the protein for protons to go from one side of the lipid bilayer to these conserved glutamate residues. The ring can rotate and bring the protonated glutamate to a second large channel through the lipid bilayer where the proton is released to the other side of the membrane. And now we know from looking at several of these structures that there's really a conserved charge distribution, where the mid-membrane terminus of these half channels has a negatively charged residue, or sometimes multiple negatively charged residues. So the protons would go actually between these alpha helices and come out over here, and the protons are released on the other side of the membrane and encounter this negatively charged residue. And at the interface of this green subunit and the ring, we have this absolutely essential arginine residue, a positive charge that prevents a shortcut of protons. It prevents protons from going from this half channel to that half channel just along here. It forces them to come along, take a ride on the ring, and then leave through their correct half channel. So once we'd done the mitochondrial ATP synthase, we reasoned we should try to get a structure of the bacterial ATP synthase, and Hui was extremely successful in doing that. So this is first with a model organism, Bacillus PS3, a thermophilic eubacterium. And due to the availability of new computational methods that let us focus the computation on one part of the structure or the other part of the structure at a time, Hui was able to get high resolution structures for the intact enzyme, even though he had still the multiple rotary states. So he could resolve each of the rotary states, and then for each rotary state, get to high enough resolution to build an atomic model. So we were very pleased with this structure. It explained about 40 or 50 years of biochemical results that people had obtained looking at mutations in bacterial ATP synthases. But then we very quickly realized that we should be looking at the mycobacterial ATP synthase because of its importance in medicine. So I already told you about the canonical electron transport chain. This is the one found in mitochondria and many bacteria. But mycobacteria, such as Mycobacterium tuberculosis that causes TB, have a rather different electron transport chain. Rather than a single complex 1, they have two forms of the enzyme, one that can pump protons and one that can't pump protons. In fact, it seems that the proton pumping form is not expressed at high levels in most conditions. And then rather than a complex 3 and complex 4 that pass electrons between them with a soluble cytochrome C, they actually have an obligatory supercomplex where the cytochrome C electron carrier is now represented by a domain that's permanently attached to the supercomplex. And they have another enzyme that can also carry out the same quinol oxidation and water oxygen reduction, but this time without pumping protons, in the cytochrome BD. And this complicated electron transfer chain allows them to adapt to a variety of energetic conditions, which makes them quite a difficult aerobic pathogen to kill, because they're very adaptable due to this flexibility of their electron transport chain. And the reason why the mycobacterial electron transport chain is so important is that there are verified, approved TB drugs that target the electron transport chain, and other TB drugs that are in clinical trial that target other parts of the electron transport chain, or this oxidative phosphorylation process. So TB, as you may know, is before COVID-19 was the leading cause of death by infectious disease internationally, and for many, many decades that has been the case. It results in about 1.4 million deaths in 2019, about 230,000 childhood deaths every year, and there's increasing drug resistance, which makes it an extremely concerning pathogen. So many of our first-line therapeutics are no longer as effective as they used to be. Our first foray into working on the mycobacterial electron transport chain was actually due to a friend and colleague, Peter Brzezinski in Stockholm University, and they had managed to purify this mycobacterial respiratory supercomplex, and we teamed up to obtain a high-resolution structure by electron microscopy. And that had some very interesting findings. It showed us that during the passage of electrons from complex 3 to complex 4, we actually detected conformational changes in the cytochrome CC domain, the thing that couples the electron transport between complex 3 and 4, and is actually essential to the proton pumping activity of the respiratory supercomplex of Mycobacterium. But the most important drug that targets the oxidative phosphorylation, this coupling of oxygen consumption to phosphorylation of ADP, the most important drug is a small molecule called bedaquiline that was discovered in 2005 at Janssen Pharmaceuticals, approved by the US FDA in 2012, and is now on the WHO list of essential medicines. And that drug, bedaquiline, is a mycobacterial ATP synthase inhibitor. Médecins Sans Frontières estimates that there should be about 400,000 people a year taking bedaquiline. Pricing has made that a number, has not allowed us to reach that number, although Janssen did recently decrease the price of bedaquiline, not to $1 a day as MSF was advocating, but I believe it's to $1.50 a day, which lets us get closer, although not quite, to the need of supplying bedaquiline to these 400,000 patients a year. And bedaquiline right now is just indicated in multi-drug resistant and extensively drug-resistant tuberculosis. Our first attempt to purify the mycobacterial ATP synthase was really just to use the trick that we had used for the Bacillus PS3 ATP synthase, and that is to take the entire operon that encodes for all the proteins that make up the ATP synthase out of Mycobacterium tuberculosis and put it into E. coli and express it and purify it there. That worked beautifully for Bacillus PS3, but it didn't actually work so well for the Mycobacterium tuberculosis ATP synthase. This is an image that my lab manager, Stephanie Buehler, obtained, where you can see in not cryo-EM, but negative stain EM, a few particles of the mycobacterial ATP synthase. We could select and average these particles and get this class average image, as we call it. But this is an image that I actually use in a grant application, and this class average is very intentionally placed right over here because it covers up a really ugly aggregate of these ATP synthase particles. And so it became clear fairly early on that this way of isolating the mycobacterial ATP synthase was not going to be effective for getting a high resolution structure. But it was effective for the grant application. So the grant was funded, we bought Stephanie a cake, and we moved on. We tried to find a new way of obtaining the mycobacterial ATP synthase. And the solution turned out to be a method that was suggested by my colleague at the University of Toronto, Jun Liu, who's a mycobacterial expert. And so what Jun suggested was using a technique called Orbit that allows us to insert a tag into the chromosomal DNA of mycobacteria. It can be used in tuberculosis, but tuberculosis is a very slow growing and of course pathogenic organism. So instead we use the closely related organism Mycobacterium smegmatis. And in fact, Mycobacterium smegmatis is the organism where this drug bedaquiline was originally identified. So smegmatis was used to screen for drugs, and that's the, we know that bedaquiline will effectively target the Mycobacterium smegmatis ATP synthase. So we knew that that enzyme was a good model for how this drug works. So now that we have a way of tagging proteins in Mycobacterium, we could purify them. And this became the project of two graduate students, Hui Guo, who at this point is getting close to graduation, and Gauthier Courbon, a new graduate student who learned from Hui's expertise in how to work with these ATP synthase complexes. So Hui and Gauthier were able to get these rather beautiful cryo-EM images of the smegmatis ATP synthase, and from that they were able to obtain this high resolution structure of the smegmatis ATP synthase. And to a first approximation, it looks an awful lot like any other bacterial ATP synthase. You can see the density map over here is good enough to build quite a reliable atomic model, but the overall structure looks very similar to the Bacillus PS3 ATP synthase. There are some differences. For example, this peripheral stalk structure, if we compare the structure in smegmatis to the structure in PS3, you can see that there are actually some rather striking differences. But if you look at the proton translocating subunit, either comparing the smegmatis structure in green to the PS3 structure in gray, you can see they're basically identical. And then there's a subsequent E. coli structure shown in purple. And again, the smegmatis structure and the E. coli structure are essentially identical. So not a very surprising ATP synthase structure. There was one thing that was peculiar about this structure initially, and that was how it prevents ATP hydrolysis. Every organism has to prevent ATP hydrolysis by their ATP synthase, or else in conditions where there is insufficient energy to build up a proton motive force, the ATP synthase, rather than making ATP, will start to hydrolyze ATP. And this is extremely deleterious to an organism. In mammals, we have a protein called inhibitory factor 1, or IF1, and that will actually bind to the ATP synthase during ischemic conditions and try to limit ATP hydrolysis. It's not too successful, and so during ischemic events, such as a stroke or a heart attack or a surgery, the ATP synthase will actually hydrolyze a significant fraction of the cell's ATP and can lead to significant cell death during something that we call ischemia reperfusion injury, where even though we reperfuse a tissue with oxygen, the hydrolysis of ATP by the ATP synthase and other factors lead to cell death. Bacteria need to be even better at preventing ATP hydrolysis because they encounter ischemic conditions far more frequently. So looking at how the Bacillus PS3 enzyme prevents ATP hydrolysis during conditions where there's no proton motive force, you can see this mauve or purple epsilon subunit actually goes up. There's an extended alpha helix that penetrates between the catalytic subunits. And if you look at the structure as a cross-section, you can see that this alpha helix in the mauve epsilon subunit is positioned so that the central rotor cannot rotate in the hydrolysis direction. If we tried to rotate the rotor in the hydrolysis direction, we would get a clash between this purple subunit and the gray beta subunit over here, but the enzyme can rotate in the synthesis direction. So we can rotate in this direction, and we have a clear space for that purple subunit, and by the time it gets over here, there's a conformational change, so now there's a space, and it can continue to rotate in the synthesis direction, but as soon as you run out of a proton motive force, you can't rotate in the opposite direction. If we look at the smegmatic structure, that alpha helix that in Bacillus PS3 penetrates between the catalytic subunits isn't long enough to reach into the catalytic region, and it's not in what we call the upward-facing conformation. It's actually in a downward-facing conformation, so it's not even trying to get in between these catalytic subunits to prevent rotation in the hydrolysis direction. However, when we measure the ATP hydrolysis activity of this enzyme, it's non-existent. So somehow this smegmatis, this mycobacterial ATP synthase, does not hydrolyze ATP. And we believe this is particularly important for mycobacteria because, as you may know, tuberculosis can persist in a dormant state, in a low energy state, for decades before infections become active. And in order to do that, the organism needs to really be very conservative with its energy usage. So it has to be able to turn off ATP hydrolysis by the ATP synthase. What we did notice is a unique extension from one of the subunits that's not seen in other ATP synthases. So this red alpha subunit has a carboxy terminal extension that extends all the way down basically to the membrane and interacts with the rotor. So this extension is seen in each one of the rotational states, and it does something that is really bad for a rotary machine. It connects the static part of the enzyme with the rotating part of the enzyme. And if we look carefully at that interaction, you can see that this extension not only interacts with the rotating part, it actually goes underneath it in a sort of hook-like conformation. And so we were new to mycobacterial genetics, and if your only tool is a hammer, all problems look like a nail. And so we decided to test this hypothesis that this extension is preventing ATP hydrolysis by doing our tagging experiment. So what we did, instead of inserting the tag up here for purification, we actually inserted the tag in the middle of the alpha subunit, or not in the middle, but just C-terminal to this to the serine residue that's the beginning of the extension. So now we replace this extension that we propose inhibits ATP hydrolysis with the affinity tag that we use to purify the enzyme. And when we purify ATP synthase that has had that truncation of this extension, we now have significant ATP hydrolysis activity. And so this, we believe, shows that truncation of the alpha subunit is sufficient to prevent, to stop the inhibition of ATP hydrolysis that allows mycobacteria to survive low energy conditions. So if you put this together into a model, you'd have this conformation of the enzyme initially, where proton translocation allows rotation in the synthesis direction. That will cause a bending back of the central rotor. But if you try to have hydrolysis, it's going to pull on this extension and prevent further rotation. So if proton translocation occurs, you do get one step of, and we did observe this alternative conformation also experimentally, you get one step of rotation that bends back this inhibitory extension, and you can continue with further proton translocation and further rotation in the synthesis direction. That will cause this extension to release. But if the proton motive force collapses, this extension can rebind and then eventually get pulled tight to prevent hydrolysis. So we can put that into a movie where you have proton translocation causes rotation in the synthesis. Well, you can't get ATP hydrolysis, but you can get proton translocation leading rotation in the synthesis direction that causes that bending back and then release of that extension. So you can continue with rotation in the synthesis direction. But should the proton motive force collapse and a rotation start to occur in the hydrolysis direction, that inhibitory extension will rebind, and then with one click in the hydrolysis direction of rotation, be pulled tight and prevent further ATP hydrolysis. So that brings us to the point where we can look at the binding of this drug, bedaquiline. And bedaquiline had a slight mystery to it, because we know that bedaquiline is a potent nanomolar inhibitor of ATP synthase. And you can see that in wild-type mycobacteria, whether it's Smegmatis or tuberculosis, you can prevent ATP synthesis with nanomolar concentrations of the drug. And we know from work done by the original groups that discovered bedaquiline, or collaborators of that group, that something about the binding site involves one of the membrane subunits of the ATP synthase. If you make a mutation in the C subunit that's part of the membrane region, you remove, these are the empty boxes, you remove the sensitivity of the ATP synthase to bedaquiline. So there's this nanomolar inhibition, and we can now recapitulate that with our ATP hydrolysis assays. We see an IC50 where we reduce the ATPase activity to 50% at nanomolar concentrations of bedaquiline. But when people have looked at the affinity of the drug to the C ring, the subunit where we know there's an interaction due to that mutation, only micromolar affinity can be detected. So a crystal structure was determined of just the isolated C ring of the mycobacterial ATP synthase with bedaquiline bound, and the affinity has been measured by multiple groups between the C ring and the drug, and only micromolar affinity can be detected. So that's a real conundrum. How can you have micromolar affinity but nanomolar inhibition? And our hypothesis is that the truly relevant binding sites for that nanomolar affinity involve not just the C subunit, but the other membrane subunit of the ATP synthase, the A subunit. And so what Hui and Gauthier did is now take the mycobacterial ATP synthase and saturate it with the drug bedaquiline and redetermine the structure. And you can see all of these binding sites. You can actually see 7 of these binding sites. There are 5 of them that just involve the C ring, and I've colored those in yellow here. We refer to those as the C-only binding sites. But there are 2 copies of the drug bound at the interface of the C ring and this green A subunit. And we refer to those as the leading binding site and the lagging binding site, depending on the direction of rotation, based on the direction of the rotation of the ring during ATP synthesis. So in the C-only site, we can see all of the interactions that were seen in the crystal structure. You can see a phenylalanine 69 residue that moves from this conformation in the drug-free structure to this conformation in the drug-bound structure. And you can see nice density for the drug in the C-only binding site. But if you look at the binding sites that also involve this green A subunit, you can see even more interactions. So at the lagging binding site, we see a deeper pocket. We still see the conformational change of this phenylalanine 69 from this conformation in drug-free to this conformation in drug-bound. And then if we look at the leading binding site, we see an extraordinarily deep binding pocket for the drug and multiple conformational changes of side chains that occur upon binding of the drug to the protein. And so it's our hypothesis that these 2 binding sites, the leading and the lagging site, are the ones that bind with higher affinity and explain the nanomolar inhibition of ATP synthase by bedaquiline. And so we could actually do a kind of cheeky experiment to test that hypothesis. We have our drug-saturated structure, and you can see the conformation, you can see the drug in all of these binding sites, C only, lagging, and leading, and you can see the density for individual side chains in these drug binding pockets. And we have our drug-free structure where you see an absence of drug density and also the conformations of the side chains, and really nice density for this tyrosine 68 residue that tells us that we have high resolution density for the binding pocket. So in our cheeky experiment, what we did is we took the drug-saturated ATP synthase and then just washed it to try to remove any loosely bound drug, with the idea that the tightly bound drug at the leading and lagging site should stay more tightly bound than the C-only sites. And if we could detect the difference of binding in those sites, we could say that these actually were the high affinity sites. That's exactly what we saw. After very mild washing conditions, you can see most of the density for the bedaquiline at the C-only site disappears. And the conformation of this phenylalanine 69 residue now more closely resembles the drug-free state than the drug-bound state, whereas density remains at quite a high level in both the leading site and the lagging site, supporting our hypothesis that these are the high affinity sites that explain the nanomolar inhibition of the mycobacterial ATP synthase by bedaquiline. So we can again combine this into a movie that shows the apo state of the enzyme, the drug-free state, and then what you'll see is that as the drug binds, we're going to zoom in on the leading site, we actually get a massive conformational change in the enzyme. The whole ring of the enzyme has to rotate by a huge rotation in order to create the drug binding state. And that rotation is remarkable in that it not only creates a leading site, it simultaneously creates just the right positioning of residues at the lagging site so that a bedaquiline molecule could be bound there. So this is really, to my mind, equivalent to what happens in evolution and natural selection, where during drug screening, you select for the molecules that are just perfectly sized and conformed, have just the right conformation in order to really match any sort of binding sites that may be available in the structure. There is one really interesting mystery that remains for us with how bedaquiline inhibits the ATP synthase, and that has to do with an uncoupling activity that bedaquilin's been shown to have. So if you look at oxygen consumption by mycobacteria, when you add bedaquiline, you actually increase respiration. It's as if you've allowed protons to pass the lipid bilayer freely so that the organism starts hydrolyzing oxygen quickly in order to try to build up its proton motive force. So this effect of adding high concentrations of bedaquiline resembles what happens if you add a conventional uncoupler, CCCP, that just can shuttle membranes, so it can shuttle protons across the membrane. You just get this uncoupled consumption of oxygen where you get very fast, a respiratory burst as it's called. And if you make a mutation in the drug binding site for bedaquiline, that C subunit that's been known to be involved for a long time, you can remove this uncoupling activity. And so there are two hypotheses for how bedaquiline can cause uncoupling. Either it can do something to distort the ATP synthase structure so that protons can flow out of the inhibited enzyme, or it can allow protons to just flow across the lipid bilayer. And so these are the two hypotheses. It either uncouples the membrane, like CCCP does, or it breaks the ATP synthase so that protons can flow through even though the ATP synthase is inhibited without making ATP, and that leads to faster respiration in the rest of the electron transport chain. And remarkably, we actually see uncoupling in the purified ATP synthase, where we see inhibition of ATP hydrolysis activity in the nanomolar concentrations, but when we get up to micromolar ATP synthase, we start to get ATP hydrolysis again, even though we know that bedaquiline is bound to the membrane region and is preventing rotation in the membrane region. So one hypothesis that Gauthier is now trying to track down is that the binding of the drug will actually break the membrane region of the ATP synthase when you get to high enough concentrations, and that actually allows protons to flow through the lipid bilayer. And it's even been proposed that this uncoupling is important for bedaquiline's bactericidal activity. So we know that bedaquiline is bacteriostatic, it prevents mycobacterial growth at nanomolar concentrations, but it has been proposed that you need micromolar concentrations to actually kill mycobacteria, and this uncoupling might be part of that activity. So we have many things to continue to look at. We want to continue to try to understand mycobacterial respiration. It's rather complicated and fascinating and a verified drug target for TB drugs. We want to understand how existing and candidate therapeutics actually interact with the respiratory chain. In order to understand mycobacterial respiration, we need to understand eukaryotic respiration better. And we also want to continue this synergistic research on the vacuolar type HPAs that I didn't really tell you about today. Underlying these three research directions are the cryo-EM methods that my lab puts a lot of energy into, because I think most of this research has been driven by the new methods that have come out in the cryo-EM community in the past 20 or 25 years that Trevor and I have worked in it. So I'll stop there. I've shown you I've told the names of all the people whose work I've described. Here's our socially distant picnic last summer, but I'd be happy to take any questions if there's any time left and any interesting questions.
Thank you, John. That was a spectacular tour de force. I must say to you that we have been watching this work with great interest, and we really are happy that you worked out how bedaquiline works. And I've asked Val Mizrahi to say one or two words. She's going to tune in and thank you to you for the talk. Val, over to you.
Yes. Thank you so much. So John, I'm not sure if you recognize my name, but I
do, yes.
Of the News and Views article.
Thank you so much. I really appreciate that.
The story there is that Trevor, late one night, I got an email from our old friend Trevor saying, "Have you seen this bioRxiv preprint? The movies are stunning." I'd seen it, but a few days later, I got contacted by Cliff Barry, who said to me, "Have you seen these movies? Do you want to write the News and Views with me?" John, this is just a spectacular piece of work. I think that we raved in the News and Views article, which was damped down. Some of our hyperbolic enthusiasm was damped down by the editors who like it to be written in a very staid kind of way. And I think the resolving of the conundrum of the binding to the C ring alone, the fact that you resolved a conundrum which is essentially going from micromolar to nanomolar, showing this beauty of cryo-EM and the ability to build these structures and these movies that show this induced fit was really one of the most appealing and satisfying things for a person like me who's worked on TB drug discovery, and in fact some of the very earliest work on elucidating the respiratory chain of mycobacteria. Our papers go back more than 25 years in this field. It was particularly exciting for us to see this. Couple of points to note. Bedaquiline, for those who don't know, was discovered in M. smegmatis in a relatively small phenotypic screen. People today don't like to screen against smegmatis. We do our screening against MTB, but who would have thought that a small compound library would deliver a molecule whose mechanism is really reminiscent, as John has highlighted, reminiscent of what you would expect natural product, where there's been a way to evolve. And yet this molecule shows this absolutely spectacular ability to go in and jam the ring so that the thing stops rotating. Besides that, the ratchet mechanism to prevent hydrolysis resolved a long-standing conundrum as to why and how does MTB conserve, or do mycobacteria conserve energy. So this was a beautiful example of structural biology coming quite a lot after the introduction of this drug, which, John, is absolutely revolutionizing the treatment of drug-resistant TB, which is a disease, a form of TB that South Africa is afflicted with as one of the worst affected countries in the world. I'm very excited to hear that you're applying your energy and your formidable intellect of yourself and your team to look at other targets in the respiratory chain. So I was wondering whether, in fact, you have any thoughts or ideas as to why the QCRB component of the BC of the super complex, the BC1, why is that such a promiscuous target where we're finding resistance mapping of so many different chemotypes, or mapping to mutations in QCRB? I think Tom Blundell and others have done modeling to try to understand why so many different molecules show resistance that maps there. But there's a big difference in the drug field between mechanisms of resistance and mechanisms of action. So I would love to know whether you are actually going to be pursuing any QCRB chemotype looking structurally at how that interacts with the BC1 AA3 supercomplex. And I'll stop there, Trevor, because I could go on for an hour just raving about this.
I think, first of all,
leave it with a question for John there. Thank you.
Thanks, Bob.
First of all, thank you so much. It's truly, Dr. Mizrahi, it's truly humbling to hear those comments. I mean, we were newcomers to the TB field, and we try to do something that we hope is important, and to hear from a pioneer such as you that it actually has an impact is really extraordinarily gratifying, and thank you so much for that, and for your news and views, which was really humbling to see. So I'm almost speechless. We are indeed looking at the respiratory supercomplex in close collaboration with Peter Brzezinski, who's extraordinarily knowledgeable about the respiratory chain. And I mean, the mycobacterial respiratory chain is really baffling because it's a strictly aerobic organism. It needs to build up a proton motive force with its respiratory chain, yet it doesn't seem to want to. It doesn't use its complex 1, which could build up a proton motive force. It's almost expressed at undetectable levels in most conditions, it seems. And it can also bypass its respiratory supercomplex with the cytochrome BD that also doesn't pump protons. So it has a way of not pumping protons, even though as a strictly aerobic organism, it needs to pump protons. So that is really hard to understand. And I presume that the cytochrome, the supercomplex of the BCC and AA3, that the complex 3 and 4 supercomplex, if you don't have a proton pumping complex 1, it's the only way the organism is pumping protons. So it becomes extremely essential for respiration. So I would expect that it is a good drug target. As you know, Q203, I'm not sure, you probably know far better than I the status of Q203 trials. I think that's probably the furthest along. But I've also heard that there are a number of compounds and also derivatives, not derivatives, but sort of metabolic products of compounds that seem to inhibit the respiratory supercomplex. And it does seem like that should be a place where you can target respiration so that as long as the organism isn't using complex 1, and I should say we might have some results obtained in the past week that show that we might actually get more complex 1 in certain growth conditions. So I think it's really unclear to me how mycobacteria are doing this. But
yeah, so a couple of some rebuttal to that. Firstly, the BC1 AA3 is not essential in mycobacteria. We showed that you could knock it out in Smegmatis years ago. So basically, this bug ramps up BD oxidase, non-proton pumping, and yet it survives. It grows very slowly. I just want to.
So does it also bump up complex 1 when you knock out?
No.
So how does it do it?
It's a great question, but I think we also need to remember that MTBs also has nitrate reductase as well. So it can use alternate electron acceptors. I have to jump off and go to another seminar now. I just wanted to say thanks, Trevor, for organizing this. It's been a huge privilege to hear. And I think listening on our YouTube channel, every member of our lab listening into you because we've talked about your paper in Journal. Congratulations and thanks everyone. Thank
you. It's very kind. It's an honor to meet you and to have this comment.
Thank you. Bye-bye.
Okay. Zoe put up a hand and she's going to ask a question.
Yes, I was very keen to know if you have any ideas or theories on how this drug may interact with the membrane itself. Have you done any microscopy with the compound in the millimolar range to see if it itself disrupts or embeds in the membrane? Could that be the mechanism?
So that is one of the proposed mechanisms for uncoupling. And we can't really see the drug with our microscopy. So the power of electron cryo microscopy is really just like crystallography with neutrons or X-rays. You need to average. And so in order to average, with neutrons and X-rays, the crystal does that for you. The crystal tells you what you need to average. With electrons, you need to see from the image what you're going to average. And so something as small as the drug interacting with the membrane, we don't have enough signal in the raw image to figure out where the drug is. So I think it's entirely possible that the drug is doing something to the membrane. I think that's actually one of the preferred hypotheses by Greg Cook in New Zealand, who's also worked extensively on this uncoupling mechanism of bedaquiline, that it doesn't look like a protonophore. It doesn't look like CCCP, but maybe it is acting as a membrane interactor to create a defect in the membrane that allows proton leak.
If it was going to make pores or actually extract some of the membrane components, I guess, yeah, you could use neutron reflectometry, of course, to study the bilayer disruption of even a model system. You can use.
That's an interesting experiment, a supported lipid bilayer looking at neutron reflection.
And then you can
a high
titrate
concentration drug.
in millimolar amounts and see if you see thinning, buckling, and or some of the lipid just being extracted by the drug.
Yep.
That's a possibility.
That's a good idea.
It's a reasonably straightforward experiment to do. And then the last question is, if you have drugs like this that may target the mycobacterium membrane, how is the membrane composition sufficiently different from the mammalian host to make it a druggable target, or is it too close?
So the hypothesis is not that it's being specifically attracted to the membrane, but because it binds the HB synthase, it accumulates in the membrane. It partitions the membrane due to the protein, and then in the membrane, it does something. So that's the model for how it can uncouple via the membrane. That it's actually it's still the high affinity binding the protein that causes it to accumulate.
Super fascinating. Thank you so much.
Thank you so much.
Manny Carisi, would you like to ask?
Yeah, if I can. First of all, congratulations on fantastic work, really.
Thank you.
I think we are running a little bit out of time, but you know the major problem with bedaquiline is cardiotoxicity. And this goes down to a potential inhibition of mitochondrial human ATP synthase. And I'm now wondering, what do you think, with a special point on uncoupling activity, so what you just mentioned, about to exploit your new binding site and your mechanism to design a second, evolution of bedaquiline? Because, bedaquiline is extremely, it's the first drug after 50 years in TB drugs. It's a terrific contribution, but it's still limited in usage to the MDR because of the cardiotoxicity. So everybody's looking for the potential evolution of the drug. Can you briefly comment on that? Thank you very much. And congratulations again.
Thank you, very, very kind. So I know there's an organization called TB Alliance that's based in South Africa and the US. They have second generation diaryl quinolines that are proposed to or reported to have lower cardiotoxicity. We've gotten some of those and we're trying to look at the drug binding side, but it's our hope that we can contribute to the medicinal chemistry effort of designing improved diaryl quinolines with these sorts of structural studies. The medicinal chemistry is pretty far from my area of expertise, but anything we can do to help, I'd be happy to do.
So you're saying that with the second generation or maybe third generation, you can increase the uncoupling activity possibly selectively,
or increase the selectivity for the mycobacterial enzyme.
Let's start with the selectivity on the target, and we look at the membrane, which will be the real target. Thank you very much. It's fantastic work. Thank you.
Thank you.
Thank you very much, John. It's been a real privilege listening to you. Thank you so much.
For me too. Thank you.
Move the conference on now. So it's Roberta's chance next, and she's going to talk to us about NMR. So, okay. So we have Roberta Pieratelli, and she's going to give us a talk on structural and unstructural biology by NMR spectroscopy. Are you ready?
Thank you. Can you see my screen?
Yes. Yes, I can see your screen, and I
cannot actually. So I have a little of a problem.
Screen mode.
Oh, where is it? I cannot see it. My sharing is post distance. Okay.
There we are.
Yeah. Sorry for that. Okay, thank you. Thank you very much for allowing me to present, to give this presentation about the use of NMR. I've seen fascinating lectures just before me, but I will try to convince you that also NMR can provide some useful information in structural biology. And I'm coming from Florence. Florence is well known for historical churches, buildings, and arts, but there is also the university, and at the University of Florence, there is the Magnetic Resonance Center, from which I'm coming. What you see here is the aerial view of the magnetic resonance center, and these numbers do represent the frequencies of the spectrometers for NMR spectroscopy that are hosted in our infrastructure. We have an array of instruments that goes from low field to high field. We have a 1.2 gigahertz that is the first commercially available instrument that's such an high field available for structural biology. We have several instruments dedicated to high resolution NMR in solution, then several instruments dedicated to solid state NMR, again at high resolution. We have other instruments dedicated to drug screening or maybe to metabonomics, to mass, HR mass and other techniques, specialized techniques. And then we have a fast field cycling relaxometer that operates at very low field, and altogether these instruments cover a very large range of frequencies from 1.2 gigahertz to the magnetic field of Earth, more or less. And of course, NMR is central to our research, but we have we have all the equipment necessary to study proteins and biological molecules alone and in their interaction, starting from bioinformatics, protein production laboratories, because we need the isotope labeling for NMR spectroscopy. Then we have systems for mammalian expression and an array of instruments for the characterization of the proteins and the systems we are interested in, including a cryo-EM spectrometer installed last year in the department nearby. And we are connected to the computing grid, the European computing grid for high performance calculation. We are the Italian Center of Instruct ERIC, and Instruct ERIC is an infrastructure, a shared, a pan-European research infrastructure dedicated to structural biology, and dedicated in particular to integrated structural biology. It is constituted by several countries that have developed the infrastructure, that are funding access to the instrumentation and to the technologies of the infrastructure. And these are mainly European countries. We have also Israel and United Kingdom that now is out of the European Union, as you know. And this is one of the infrastructure dedicated to life sciences that has been established in Europe thanks to the life science research infrastructure initiatives of the European Commission. And the infrastructure is open for access to the instrumentation, to also activities in training. And it is not only open to the funding countries. It is open also to scientists that are coming from other countries, and there are every now and then programs for allowing scientists coming from other countries to access the instrumentation. For example, there was a call for South American scientists. We have access from other continents as well. And this is one opportunity to come and use our infrastructures. And that covers many different aspects of structural biology that goes from sample preparation, including crystallization, protein production of different types, nanobody discovery, and then has platforms for biomolecular analysis, including imaging, mass spectrometry, molecular biophysics, and whatever you may need. And also 3D structural analysis. We are, of course, central to the activities of the magnetic resonance platform, but there are also electron microscopy and X-ray facilities dedicated to structural biology. We are also part of a network of infrastructures linked in the project iNext Discovery, that is an European funded activity. And again, this is an action that is targeted to scientists to have access to give them access to the instrumentation, and also to develop projects, specific research project dedicated to improve the quality and the quantity of access, and also networking activities, training activities, and so on. And this is open to, it is mainly European, as I said, but it is open also for researchers from other countries. We have about 30% of the access activity to our instrumentation that is for scientists coming from third countries. So Africa, South America, United States, Australia, whatever you think. And it is particularly important, I think, for non-expert in structural biology to get access to such a facility, because we provide also a training. We have the equipment, we have a personnel that is dedicated to the support of the users, both in the wet lab and in for the spectrometers in our case, but also for the other techniques, for the other to the other facilities. And we have a scientific expert supervision that is provided by the staff of the academic staff of our center. In particular, we are all faculties of the University of Florence, and we are there. We are here also to promote the use of this facility and the NMR spectroscopy in particular, that, as you can imagine, is central for us. And this little drawing, we have a cell, very simple description. And what you have here are the little structures of the proteins that we have solved in the last few years. And in particular, these are structures related to the homeostasis of the copper ion in the cell. And starting from designing the structure, resolving the structure of these molecules, you can, by NMR, you can study protein-protein interactions, for example, particularly when these interactions are transient and so difficult maybe to address by other techniques. And then you can also map the interaction of a small molecule of the protein with small molecules, like, for example, in this little drawing, you have an NMR spectrum, and the blue dots represent some specific signals. And then when you have a little molecule that goes interact with your protein, you have a different set of signals, and the movement and the different positions of the peaks immediately tell us something about the location of the drug and what are the changes in the structure and in dynamics that you have when you have this interaction. And of course, if you have an eye field, you can really map precisely your molecule also in a simple 2D experiment like this one that is correlating proton and nitrogen. Here you have a protein related to the SARS-CoV-2 virus, and it has been assigned as many other proteins of the SARS-CoV-2. And by the way, this is the occasion for me to tell you about this initiative that is a worldwide initiative that is a project linking together many labs working towards the assignment and the characterizations by NMR of the proteins of the SARS-CoV-2, and all the targets are now being studied and solved. If you are interested, you can go and look at this site, where you can also join, if you like, to contribute to these efforts toward in our fights against the coronavirus. NMR spectroscopy is now developed at the point in which you can study directly in the cell your proteins. You can express and immediately study your proteins either in Escherichia coli or in mammalian cells, in particular very relevant for some studies about function of proteins, and then monitor in the cells directly protein folding, aggregation, maturation, and so on. You can study the occurrence of post-translational modifications, for example, like the establishment of some disulfide bridge or some phosphorylation or some other happenings. in real time, more or less. And then you can study redox dependence, folding, regulation, and so on. And you can arrive to study also drug interactions in cell directly. For example, this is the case of a protein that has been mentioned by Zoe, that is a carbonic anhydrase, that is a pretty protein where zinc metallobinding protein, where you have 3 histidines, and these are the signals, the NMR signals of these histidines in the cell, as you can monitor them directly when the protein is still in the cell. And you can see, for example, upon interaction with the various sulfonamides, how the signals move, how the signals change position. And this can be, this is very important because you can monitor the interactions, the occurrence of the interaction with the sulfonamide, and you can also monitor the kinetics of the process and the thermodynamics of the process, and for example, immediately assess drug potency and the capability of a drug to enter the cell and to directly interact with the protein of interest, and so on. You can use NMR also in the solid state. You can have crystals. Often you use deuterated crystals, as Zoe tells us, it's very important to be able to deuterate your protein, and you can get the structure in microcrystalline form. But what is most important, it is that you can study the dynamics of the system in the solid state that enable you to access motional regimes that are not easily accessible by other means. For example, here we have SOD, superoxide dismutase, that is our PET protein in the lab, and we can monitor the uptake of the metal ions because this is a metal binding protein. And for example, you can monitor the variation of dynamics of the ligands upon interaction with the copper ion. That is, yes, important. But what is most important is that you can monitor a variation in dynamics very far from the active site. And these are very relevant for function because they are somehow related to the capability of this protein to dimerize and to be functional in the dimeric form. And then you can study the protein when it is when they are grafted onto Galton particles. You can have very different approaches. But what is more, I think, interesting to this audience is the fact that you can study the dynamics of the system, and you can study highly dynamic systems. Proteins. Indeed, you know that next to the well-folded proteins, you do have proteins that are completely disordered, but are nevertheless functional. And in between these two extremes, there are all the possible states that are, for example, proteins that have secondary structural elements but are in a very dynamic environment, or proteins that are not structured but get some structural elements when you they interact with a molecule or a partner or and so on. And disorder is quite abundant in the proteome. And for example, it's been established by bioinformatics that about 45% of the human proteome contains proteins that have stretches of amino acids that are disordered, and they can be very long disordered stretches. And indeed, there is advantages in being disordered because we have plasticity, malleability of the chain that enable the establishment of multiple interactions, and also to expose part of the polypeptide for post-translational modifications, or to establish interactions even in the same chain, in the same peptidic chains with many different partners. So it is very important, and indeed the disorder is involved in many processes like molecular recognition, molecular assembly, transcription, regulation, and so on. And when it comes to study systems with no stable 3D structure and very dynamics, NMR spectroscopy is, of course, a very useful resource, and particularly at high field. Because when you do not have a structure, you have a spectra that are very crowded. You see here we have the spectrum of SOD, the protein I was mentioning before, where you have proton-nitrogen correlations, peaks, that reports about every single NH, peptidic NH in your protein, and you see that these are very, very well dispersed. Whereas when you have alpha-synuclein, that is a typical disordered protein, you have that all the resonances, all the signals are really overlapping in a couple of, in a 1 ppm or less. So you need resolution to solve this. And in Florence, we have established several approaches also to overcome this resolution problem. And one is, for example, to exploit the next to proton detection NMR, carbon detection of NMR that you can see from this plot provides more resolution. And then we have implemented all the tricks to obtain the spectra in a reasonable amount of time, reducing acquisition time, still maintaining high resolution. And we are also involved, very, very active in developing experiments. That seems complicated, but they are not, because they can be done just pushing a button when you have established the procedures. And just to give you an idea of what we can do, I'll tell you about a little story about the linkers of one of the linkers of the Creb binding protein. The Creb binding protein is a transcriptional co-regulator that is capable of doing many different activities. It is a quite a large protein, 2,400 residues, and it is mainly a histone acetyltransferase protein, but has many regulatory functions, and you have these well-folded domains that are several zinc binding domains like TADS1, TADS2, the PHD, the ZZ, and then there is this HAT domain, a bromo domain that recognize acetylated lysines and so on. There are more than 400 partner proteins that has been identified, and the domains, the structured domains, have been all characterized either by X-ray crystallography or NMR spectroscopy or both. So it is a quite well-studied system. But often when you want to study a folded domain, you chop the disordered linkers of such a large proteins. But these linkers do constitute about 60% of the amino acid sequence of this protein. And they are not so innocent. For example, if you have a phosphorylation of a serine that is located in the first disordered linker, the ID1 at the N-terminus, you have an inhibition of the HAT activity that is very far in sequence and completely, it seems completely unrelated in function. So they are there for something. And together with Peter Tompa, we in Brussels, we started a systematic investigation of these linkers. And I will tell you now about one of these linkers, the ID4. That is the linker connecting the TADS2 domain and the nuclear coactivator binding domain. You see here the sequence of these 200 amino acid protein, and you can recognize some of the typical amino acid composition of an intrinsically disordered proteins. You see that there are many Qs, there are many Ps, there are many arginine. All these arginine, serine, glutamine, proline are all typical disordered promoting residues. So when you have a large share of this amino acid, you can expect a protein that is not going to fold easily. And we can measure a spectrum, an NMR spectrum. This is a simple one. And with carbon detection NMR, you can even map the 45 prolines that are present in this sequence that constitute about 22% of the sequence. That is a lot if you think and stand usual composition of the protein. And with the combination of one proton detected and carbon detected multidimensional NMR experiment, you can assign, so you can identify all the nuclei that are present in the protein and link each amino acid after the other and have a complete picture of this protein if it is completely disordered. And what you see when you have the assignment, you can start measuring NMR observables. There are many that you can measure and that can tell you about the characteristics from the structural and dynamic point of view about your protein. I just report here a few. And these are, for example, the heteronuclear NOE, that is a clear measure of the disorder of the dynamics of your system. For example, when these values, these histograms are close to 0, you have an highly dynamic system. And you look at this and you see that most are very small, but there are two regions, these two regions marked in blue, where you have slightly higher values that are indicating that you have more structure here, less dynamic system. And also, for example, the transverse relaxation rates tell us that there are parts of the protein that act as a very dynamic system and two other regions that are a bit less dynamics. Temperature dependence of the chemical shift tell us, for example, that the system is highly exchangeable with the solvent, but this region is somehow less exposed to variation of temperature and so on. So there are several considerations that you can do, and particularly you can measure the from the chemical shift the variation of the chemical shift in your system, your experimental chemical shift, with respect to the chemical shift of the same amino acid in a random coil sequence. And these tell us about a structural propensity that is present in your protein, despite the fact that it is disordered. So you have that, for example, for ID4, you have that most of the sequence is has values close to 0, so it is really random coil, more or less indistinguishable with respect to random coil value. But in these two regions, where the values of the deviation of the chemical shift are such that indicate the presence of an alpha helical propensity. And if you look at the distribution of the prolines, for example, you see that these amino acids are all in the regions where we have random coil conformation. But the regions where we have alpha propensity, there are no prolines. And we have a number of positive residues that are distributed all along the sequence and negative residues that are present only next to the alpha helical region. So despite the disorder, there are two regions of CBP-ID4 that have a propensity to sample the helical conformation, and this could very well be molecular recognition motifs that are used for interacting during the function of this protein. We were more interested in mapping the interaction instead with an oncogenic protein, that is the A1A from the adenovirus. That is a virus that is known to target CBP to interfere with the activity of CBP and to adjust self-regulation and then overcome the control of the cell and promote the diffusion of the infection. And A1A is a molecular hub in the sense that has the capability to interact with many different proteins. And it is known that A1A interact with the TAZ2 domain, one of the zinc fingers that I mentioned before, and it is known to interact with the nuclear coactivator binding domain. And these two domains are folded domains next to ID4. So if we go to look at ID4, it is 207 amino acids, and then we have at the two extreme TAZ2 and NCBD. And the interaction of these two these two domains with E1A has been mapped in part, partially. In particular, this has been studied with a fragment of E1A. And the affinity is not that bad, but you should keep into account that all these domains are interacting with their physiological partners. So E1A should interact in a very efficient way with these domains to overcome the physiological interactors. And we started characterizing also this protein, this A1A, and it is again a disordered protein. You see from this spectrum that is all the resonances are collapsed in a small portion of the spectrum, and you see the composition is again rich in many polar amino acids, and it is rich in prolines again. So you expect also for this protein to be a disorder, and indeed it is. You see that the secondary structural propensity plot tells us that it is random coil more or less, so very less structural elements. In the beginning of the polypeptide, there is a region that is somehow showing some propensity to adopt an alpha helical conformation. Again, prolines are distributed all along the sequence and not in this little beginning of the polypeptide. We have a high number of negative charges in this amino acid sequence. And this is one of the possible conformation of this protein. Here there are others, but you see you can have really many different, you have an ensemble of conformation that are necessary to describe your system. And if you you study the interaction of A1A with the ID4, you see that there are some movements in your spectra. The black is the free A1A spectrum, and then you add increasing amount of ID4, and you see that some resonances disappear, some resonances move. Here you have a close-up of one little region of the spectrum, and you see here we have movement, here we have disappearance, and so on. And if you go to measure the intensity of the peaks with and without the E1A, without CBP, you can see which are the residues most affected by the interaction. And you can also measure the chemical shift variation to map the chemical shift perturbation map of your protein, and establish that there are 3 regions that are mostly affected by the interaction with ID4. And then you can do the job from the other side. So you can take the spectrum of ID4 alone, and then you can add increased amount of A1A, and then look at the variation in your spectra, and then map the changes, again, in this case, monitoring the disappearance of the peaks. And you can do this also with carbon direct detection experiment that is very sensitive. And then you can really establish which are the resonances that are mostly affected. And you can see that these are the residues of the first helical region of ID4, whereas the rest is not affected at all. So you have a precise site of interaction between ID4 and A1A. And there is the structure of the NMR structure of the TALS2 domain solved with a little peptide representing E1A. And it's been established that this region is the one that is important for the interaction with A1A. But how it goes that the rest of the protein, this is just a little portion of A1A. And what we can do is just to do a little cartoon. This is just for didactical reasons, let's say. It's not really meant to be really an interpretation of what we we are observing by NMR spectroscopy, but I'll try just to give you a flavor of what you can learn. This is the structure of the TADS2 domain. This is the CBP ID4. And then you can think that when A1A approach your protein and should go here to really interact with the TADS2, just come close to this portion of CBP that I remind you, it is a very large protein and it extends all over the place, and then goes next to the helical region, the first helical region with the helical region of A1A to start establishing the interaction that will bring the rest of the polypeptide, that is 245 amino acids, to wrap up around the folded domain and then help interfering with the normal function of CBP. And this is just one of the possible interpretation of the data that are corroborated by the biochemical measurements and also from the outcome of other techniques. And I hope that with this I give you a flavor of what can be done with NMR spectroscopy. And I remind you that you can give a look to our website to look at the possible opportunities for access to our instrumentation and to exploit by yourself also NMR spectroscopy. And with this, I thank you for your attention.
Thank you very much, Roberta. There is a question. In fact, there are two questions now. Cinta, do you want to ask your question first?
Excuse me?
Hello.
Hi.
Hello, thank you very much. I just wanted to know, because I keep seeing this map of secondary structure property versus residue number, and I wanted to know how it is generated. Like, is it based on chemical sheets or?
Yes. Yes, you need to assign, of course, you need to assign your protein. And taking the chemical shift of carbonyl, carbon alpha, NHs, and nitrogen, you can compare your chemical shift, experimental chemical shift, with the chemical shift that are, let's say, either calculated or determined by experimental methods for random coil protein. That is the same amino acid in the context of your sequence, but in a random coil fashion. Of course, it is not so straightforward to have a proper reference for random coil, but there are several approaches that has been established to obtain these numbers in a reliable way. And then you can compare your chemical shift with those, and then you can have an indication if your polypeptide has a tendency to assume a certain conformation, either beta strand or alpha helical conformation, or if it is instead completely random.
Thank you, Roberta. I found that talk extremely interesting, and it's opened my eyes to the possibilities of working with NMR. That I really didn't appreciate all the complexity and the information content in the spectra. Unfortunately, we have two more speakers, and in fairness to them, I'm going to have to move on. I'm sure there are a lot of people with more questions, and I'd like you to perhaps write to Roberta. People can write to you, Roberta, can they?
Absolutely.
And also you can put questions in the chat that Roberta can pick up privately with you if they're pressing questions. So yeah. Thank you very much, Roberta. And the next person. Thank you so much. And then the next person to talk is Clifford Ntui from Pretoria, and he's going to talk to us about structural and functional characterization of the secreted adhesion ETPA of enterotoxigenic E. coli. Okay, are you ready to share, Clifford? Okay, I see you've done it. Okay, over to you. Your floor is yours.
Thank you, Prof. My name is Clifford Tree, originally from Cameroon, and I've been doing my studies here in South Africa. I'm currently doing my PhD in biochemistry at the University of Pretoria. I just want to use this opportunity to thank the organizing committee for giving me this platform to present some of the work that I've done for my PhD. My talk is titled Structural and Functional Characterization of the Secreted Addition ETPA of Enterosogenic E. coli. And it has been under the supervision of Prof. Mubrisa Schubert from the Structural Biology of Infectious Disease, University of Pretoria. Enterosogenic E. coli, otherwise known as ETEC, is a common cause of fetal diarrhea in children, and colonization factors produced by this particular bacteria allow it to attach itself to epithelial lining of the intestine, which is able to produce both heat-stable or heat-labile toxins. Bacterial type 5B secretion system, otherwise known as the two-partner secretion system, is a common secretion pathway involved in virulent in some gram-negative bacteria, and this consists of two important proteins. The first one is the beta-barrel transporter, otherwise known as the TPSB protein. And the second one is the passenger protein, otherwise known as the TPSA protein. And the function of the TPSB protein is actually to transport the passenger protein from the periplasm into the outer membrane. An association between this protein lies at the level of two important conserved domains known as the polypeptide transport association domain at the TPS protein, as well as the two-partner secretion domain, otherwise known as the TPS domain at the level of the TPSA proteins. And both proteins are being secreted into the periplasm via the SEC machinery. For ATPA, is actually a secreted adenosine that belongs to the TPSA protein of the two-partner secretion system. And it has been shown to link flagellin at the tip of flagella to host cell surface. And here is actually the domain organization as far as the ETPA protein from ETEC is concerned. You have an N-terminal signal peptide, followed by the two-partner secretion domain, which is conserved among these particular proteins. At the C-terminal domain level, you have 4 repeats of about 228 amino acids in length, and this is followed by the tail fragment. So in order for us to actually study this particular high molecular protein, we decided to fragment the protein into 2 domains, with the first domain consisting of the TPS domain, in addition to extra residues of the N-terminal region, of course, without the signal peptide. The second domain, which we term the C-terminal fragment, whereby we decide to remove 3 of those repeats, such that we generated a fragment which contained 1 of the repeats in addition to the tail fragment, and decide to do our structural and functional studies on this particular fragments. So for the first fragment, which is the N-terminal fragment, we're able to produce this 38 kilodalton protein and purify it successfully. And crystallization experiment produced these beautiful flat crystals, and here is actually the X-ray diffraction pattern that we obtained from the set crystals. That was followed by data collection and refinement analysis. More importantly, we obtained a space group of P1 for this particular protein crystal, and of another important, the completeness of just above 95%. It was very, very difficult to actually complete a complete data set for this particular space group as far as this particular protein crystals were concerned. Then this is actually what defines the asymmetric unit of the N-terminal fragment of ETPA protein from ETEC. We actually observed 4 chains within the asymmetric unit, all of which showed more of a beta helical structure. And a more detail of the analysis as far as the N-terminal fragment structure is concerned, just at the level of the side view, you find out that 3 beta sheets actually constitute this particular structure, thereby forming more of a beta helical structure. And more importantly, at the level of the end view, you have 2 beta strand B1 and B2, which are known to cap the N-terminal region, thereby protecting the core of the helix from solving. And we find out that in most beta helix structure, we find out that the N-terminal is actually capped by alpha helices, but in the case of ATP, it's being capped by a beta strand. Here's just a ball and stick view of the coil formed between beta strand 9, 10, and 11. I choose to represent this particular beta strand because they actually contain one of the conserved motifs, the NPNG loop. And we also see some of the hydrogen bonds involved in the stabilizing these strands. Beta 10s are also indicated. Furthermore, we went to compare the structure of the N-terminal region of ATPase containing the TPS domain with the structure of existing TPS domain structures. So the first one is our ATPase N-terminal fragment structure, followed by the HMW1 from Haemophilus influenza, which is also an iacin involved in adherence. Then you have the HXUA from Haemophilus influenza, which is involved in ion acquisition. Then you have the filamentous hemagglutinin from Bordetella epistosis, which is also involved in adherence. Then you have the one HPM8265 from Proteus mirabilis, which is more hemolysin. More importantly, we find out that the overall fold of the beta helix is conserved among all these TPS structures. And also the ones in green are actually some of those motifs that are also conserved among this particular TPS domain. At the level of the end view, just want to point out in red, the beta strand actually protect the N-terminal region from a solvent. We call them the capping beta strands. We look at them closely, find out that the first two, including that of ATP, actually have the beta strand oriented in such a way that you have more of an open cap region, whereas the last two, you have the beta strand position such that you have more of a closed capping regions. The implication of these different capping regions is not quite well understood. Also, we also went ahead and do a more of a sequence amino acid alignment of the different structures of the TPS domain that have been solved. So here you have the conserved NPNG and the other conserved motif. I just want to point out that if you look at the conservation of the residues, there's a very low sequence identity at the level of the amino acid sequence. So when you look at the superposition of the ETPA fragment from of the fragment of the TPS domain of ETPA and that of the high molecular weight one from Haemophilus influenza, they actually align very well, indicating that irrespective of the low sequence amino acid sequence, the core of the beta helix is well conserved. We then went further to model the C-terminal fragment of ATPase because we are still struggling with a crystallization experiment. So here is actually the profile of the protein, the purified protein, which is 72 kilodalton. And if we look at the model structure, it actually shows that the C-terminal actually also maintains a beta helix structure, same as the one we have observed for the N-terminal fragment, and also the A region maintain more of a alpha helical structure, indicating that the overall fold of the ATPase structure is more of a continuous beta helical structure. Then we went further to do some CD analysis after unfolding and refolding the two domains with both thermal and chemical unfold using urea. A is actually the CD spectra obtained from the N-terminal fragment, and you can observe a minima at about 220 nanometers. Then B is actually the plot of the curve obtained after thermal unfolding of the N-terminal domain. As you can see, the protein could still maintain its folds at up to about 50 degrees, and it gradually started to unfold at about 60 degrees and was completely unfolded after 80 degrees Celsius. And C is actually a melting plot of the different temperatures at about 220 nanometers. So you can see the melting temperature for this particular domain is just about 53 degrees Celsius. And this is actually a plot of the plot showing the refolded protein after treatment with different urea concentrations. So we would actually unfolded the protein with different urea concentration, then we try to refold it and see if the protein will regain its folds or not. Surprisingly, we were able to see that even at the urea concentration of 6 molar, the protein could still regain its native fold after unfolding and refolding for the N-terminal fragment. This is actually the CD spectra obtained for the C-terminal fragment with a minima at about 220 nanometers. And figure F is actually the plot of the melting curve after thermal unfolding for the C-terminal domain. Similar to the N-terminal domain, the protein was still stable at up to about 50 degrees and start to lose its fold after 60 degrees and was completely unfolded at about 80 degrees Celsius. And a plot of the melting curve at about 220 nanometers also indicated that the melting temperature for this particular domain is also about 55 degrees Celsius, similar to that of the N-terminal fragment. We now went to investigate the interaction of these different two domains with flagellin, as previously showed by Dr. James Frankenstein and his group, that ETP actually associate or interact with flagellin. So to do this, we individually investigated the interaction of the N-terminals with flagellin. So you see this is a Histac flagellin, followed by the N-terminal, untapped N-terminal domain. It was produced as a GST fusion protein, so we clip up the GST tact. And here's the flow through after coupling both proteins together. Then after extensive wash, we're able to analyze the nickel resin to see if there will be any interaction between the Histac flagellin and N-terminal ATPase fragment. Unfortunately, we couldn't find any N-terminal ATPase fragment after analysis of the experimental fraction, indicating that the N-terminal ATPase fragment couldn't interact with flagellin. And we did the same experiment for the C-terminal ATPase fragment. So here you have the untapped flagellin, which was produced as a GST fusion protein, after which we clip off the GST tag. Then you have the Histac C-terminal ETPase domain. After coupling both proteins together, we collected the flow through, and after extensive watch, we analyzed the resins to see if there will be any flagellin that will be bound on the resins as a result of interaction with the Histac C-terminal ETPase domain. Unfortunately, we couldn't see any of the flagellin at after analyzing the experimental fragment. Also an indication that there is no interaction between N-terminal ETPA fragment and between the C-terminal ETPA fragment and flagellin. In summary, we were able to separate the N and C-terminal fragment of ETPA from ATEC. We were able to solve the crystal structure of the N-terminal fragment and also model the structure of the C-terminal region. Pull-down acid do not support any interaction of either fragment with the flagellin. And ETPA appears to constitute a continuous beta helix based on the observation of the crystal structure of the N-terminal domain and the model structure of the C-terminal fragment. CD spectroscopy indicated similar melting temperature for both N and C-terminal fragments, and more importantly, the N-terminal domain unfold and fold reversibly. That's all I have for you. Thank you.
Thank you, Clifford. Extremely interesting talk. I'm intrigued about the refolding capability of these things. That was the first thing that struck me, was that they were so robust to unmelting and refolding. Perhaps why do you think that happens? What are the nature? What's going on?
So, Prof, for the N-terminal domain, first of all, the reason we did this particular experiment, remember the TPA proteins, they are secreted. The question is, how do they unfold, or how do they fold after being transported out of the periplasm? And a number of proposals have been put in place, one of which is that the N-terminal fragment actually folds the C-terminal domain after being exposed to the surface first. And we've also suggested that if the N-terminal domain can actually fold the C-terminal domain, so it should be able to fold or to regain its fold after being unfolded with any kind of chemical, especially in our case, in the case of urea.
Okay, thank you. Are there any other questions for Clifford? I don't see any hands up. Anybody want to put their hand up? Okay, with that then, Clifford, thank you very much. It was a very interesting talk and very well prepared. Thank you so much.
Thank you, Prof.
The last speaker of the day is Dauda Traore. Have you had your screen sharing enabled for you? Dauda, can you speak?
Not yet. It hasn't been enabled, but I'm here and I'm.
Lawrence, can you enable the screen sharing for Dauda?
Done.
Yeah, perfect.
Okay, so Dauda is going to. Oh, I've lost the talk title. Dauda is going to talk to us about the molecular basis for the transfer of large toxin plasmids in Clostridium perfringens. And I must just say that Dauda has now got a job in Grenoble. So he's at the center of world structural biology. And we look forward to hearing his talk. Okay, thank you, Dauda. Over to you.
Thank you very, very much, Trevor. So the title of my presentation is going to be on my current research project, which is aiming to investigate the molecular basis for the large toxin plasmids in Clostridium perfringens. But just before I dig into that subject, so 2 years ago, I moved to Grenoble, and I am basically located at the ILL, and then the reactor dome is just behind me. So, but currently, I am a lecturer at Kiel University, but I hold a joint appointment with the ILL. So I am permanently based in Grenoble. In fact, so part of my job is to provide cryo-EM access to the neutron users, but but my job is not limited to those users. So I also serve as a user support on the Titan Cryos. So the Titan Cryos is installed at the ESRF, and we are basically located right there. And the Titan Cryos is operated by the 4 institute of the campus here. So the ESRF, of course, EMBL, ILL, and the IBS. So the IBS is the French Institute for Structural Biology. So this is a very special campus, and I am very fortunate to be located on this site. And if anyone has any question into accessing the Cryos facility or any other facilities located on the campus, so do not hesitate to get in touch. Okay, so now back to my presentation. So we are interested in horizontal gene transfer. So horizontal gene transfer is the movement of DNA, so from one bacterium to the other. So this process takes so it requires a cell-to-cell contact, and as you can see here, so one strand of the DNA is transferred into a recipient cell. And this recipient cell in turn can also become a donor cell. So you can here appreciate that this is a mechanism that is efficient in order to spread around antibiotic and antibiotic resistant genes, so, and as well as toxin gene. And this is one of the reason for the rise of the so-called superbugs. Okay, so and the aim of my research is to look at what is happening here at a molecular level. So for that purpose, so we are using Clostridium perfringens as model system. So it is a gram-positive spore forming bacillus, and it is responsible for a variety of disease. And these diseases are often toxin mediated, and Clostridium encodes more than 20 extracellular toxins and enzymes. And the majority of these plasmids are basically located on plasmids. Okay, so and those plasmids have been shown to be conjugative, so which means that they have the ability to move themselves from one bacterium to the other. And in this picture, so this is the sequence alignment here of some of those plasmids. And what we discovered is that all known conjugative plasmids in perfringens, they do have a common backbone. And this common backbone here is what they call the TCP locus here. So colored here in red. And this TCP locus, so if I zoom onto the locus, so this locus here is a cluster of genes that basically assemble the complete machinery that is necessary to transfer the plasmid from one cell to the other. So This is a pictogram of our current working model. So all of the genes that I have represented here with a tick underneath them have been shown to be important or essential for conjugation. And this model here has been built on years of molecular genetics and biochemistry studies. So whenever you see here two molecules touching or interacting, is that we have some kind of evidence that they indeed interact. So, for example, the reason why we don't know TCPD and TCPE and F are interacting with the rest of the system is we simply do not know yet how they are involved in the system. But overall, we know that the genes or the molecules are important for conjugation. And this is just some of the roles of this protein. So, and okay, so overall, so the ultimate goal of this project is to get a molecular picture of this transfer system. And when we started this, so the strategy was to divide and conquer and then try to get individual structures of the components. And due to the time constraints, so I will only touch upon two proteins today. So the first one is going to be TCPK. So TCPK is part of the relaxosome complex. So the relaxosome is where the plasmid is recognized and processed prior to the transfer. Okay, and then one thing I haven't mentioned is the ORIT. So the ORIT here, it stands for origin of transfer, and then this is where, and this region is critical because this is where the plasmid is recognized by TCPK and TCPM, processed, and then are brought here to the transfer complex. Okay, so why TCPK? So TCPK was particularly interesting because when we started working on it, so all we had was that based on the sequence analysis, TCPK was a protein of unknown function. So we went on and expressed and purified the protein, and then we discovered that it was a dimer in solution, and we determined its crystal structure by experimental phasing. Okay, and the take-home message from this structure was that TCPK was both a dimer in the crystal and in solution. And then we went back and then we took this structure and did a homology search, and then the top result was this protein here. So SLY-A, and SLY-A contains a winged helix domain, and this WHT and this WHTH domain is simply the signature of a DNA binding protein. And then that was kind of assuring in the sense that TCPK is located right next to the ORIT site. So we then decided to investigate how this molecule could bind DNA, and then the technique we choose for that is surface plasmon resonance, and then we divided the ORIT region into short fragment, and then we tested the binding of TCPK against each fragment. And this is just summarizing some of the result here. So we had a strong affinity for the first fragment, a slightly lower affinity for the second fragment, and no binding was observed for the third fragment. And then in order to do a finer screening, so we combined the first and second fragment, and then we did shorter truncations to the binding site, and then we discovered that this oligo here was the minimum sequence that is required for TCPK binding. Okay, and naturally, so we took that sequence and we used it in co-crystallization experiment, and as easy as that, we were able to get the co-crystal structure of TCPK with the sequence we just identified with the SPR experiment. And this was quite surprising, because what we discovered here was that it was an unusual DNA binding mode for a winged helix-tone helix. And we also discovered that TCPK was not binding onto one single fragment, but it was rather bridging together two separate strands. And as you can see in this picture here, so TCPK is basically bridging together two separate DNA fragment. And obviously we validated the interactions that we see here with the molecule, both in vitro and in vivo. Okay, so I invite you to look at this paper if you would like to see a bit more into the structure and the validation process. Okay, so this was TCPK, and then the final and the second protein I would like to present is TCPA, so the coupling protein. So it's called a coupling protein because this is the protein that couples the relaxosome complex with the core of the machinery. And this is some unpublished work, and I hope to submit this within the next few weeks. Okay, so getting the crystal structure of this protein was extremely difficult. So we had to go through a series of truncation. So especially at the N terminal. So because at the N terminal here, so we have transmembrane helices, and the C terminus was also predicted to be unstructured or disordered. So this is the crystal structure of the construct 103 to 360, and this was again obtained by experimental phasing. And once we had that first structure, so we have been able to get additional structures. So in particular with ATP and ATP gamma S, and we also have the structure of the APO form, and also with ADP. So overall, so what all those the structure teach us is that basically what is happening here is that we have a independent movement of the N-terminal domain, and the C-terminal domain. Okay, so and then these two domains are linked by this loop region here between the two domain, and this movement will become relevant for activity or translocation in the context of translocation on the next slide. So, and then we were able to get an additional structure, which again does not contain the N-terminal domain. So what we put in the crystallization experiment is the construct from 103 to 530, and in this construct crystallized in a hexagonal crystal form, but unfortunately residues 460s to 530 again, so they remain remain invisible in the crystal structure. So we are now trying to find ways to crystallize the full length protein, or in complex with the relaxosome component. So in order to possibly stabilize that flexible domain. So back to that examic structure. So this is the pore, and this is where we think that the DNA would transit through. So from the relaxosome component at the bottom here to the transfer complex at the top here. But this is a work in progress, and we have to try to get the co-crystal structure of TCPA and TCPH or additional partners in the system. Okay, so this is just my summary slide. So I'm just going to recap all the structures that we have done on this system to date. So we have the crystal structure of TCPC, but I haven't presented that structure today because this is a oldish project result. So what I presented today was the crystal structure of TCPK and the crystal structure of TCPA. But we have some preliminary EM data on TCPF, so which again is also a hexameric ATPase, and you can kind of appreciate that from the 2D classification here. But what we notice here is a high flexibility in some of the region of the protein. So this is why we are struggling to get a high resolution structure of this protein. But it's a work in progress, and we are also trying to get protein complexes that will be used in predominantly a cryo-EM experiment. All right, so this is just some acknowledgement. So when I started this project, so I was based in Australia, so at the Monash Biomedicine Discovery Institute, but these days I am located at the ILL and Keele University. But my main basis is in Grenoble here, and the data was collected at the Australian Synchrotron, and the research was mainly funded by the Australian government. And now I am continuing the project with some seed funding from Instruct. Okay, and this is it for my talk, and I will be happy to take any question. Thanks a lot.
Thank you, Dawood. That was an extremely interesting talk. I must say that the talks we've had today have been spectacularly interesting. I found myself gripped by everybody's talk, and it was wonderful to hear everybody's experience and to show how relevant structural biology is to the continent of Africa, and how many Africans are involved in producing results in this area. So I wonder if there are any questions specifically for Daouda. And okay, so I'm going. Are there no questions? Okay, I'm sorry about that. So I'd like to end off by thanking all of the speakers in the whole day's session. We've had really had a remarkable collection, and we've learnt about the breadth and scope of technologies that are being used in structural biology and in structural biology experiments. We've learnt a great deal about the depth and scope of the type of investigations that people are doing into various different systems, both at the chemical level, the drug development level, at the biological level, but the disease level. I mean, it has been a smorgasbord and a tour de force. So thank you so much for everybody who has participated in the session. I really appreciate it. And thank you very much to Chart and Lawrence for putting together such a superb program, and their enthusiasm for keeping this event on track, which I think has been a great contribution to Biophysics Week. It's demonstrated that Africa also has biophysics to tell people about, and I'm actually very proud to be associated with this event. So thank you very much to everybody.
And thank you as well to Emmanuel and the entire BioStruct Africa team.
Sorry, I'm sorry about that. Yes, thank you to Emmanuel and the entire BioStruct Africa team. It's been a wonderful event, and thank you so much for your talks.
All right, so that ends today. Then we start tomorrow with more talks at 1000 hours GMT +2. So that's South Africa time, Central Africa time.
Okay, thanks again. Thanks to everybody.
All right, thanks everyone.
Thank you. To you. See you tomorrow.
Thanks, Roberto.
So Trevor, I have a thought for you.
Okay, I'm here. I've got a different picture this certain.
I think, you know, one thing we should do is try to collect what facilities people have locally, no matter what state that they are in, and then figure out how each of those facilities might plug into like Instruct. That's point one. And the other is what investments would those facilities take to sort of operate at a very high level for the next 5 years?
I think that's a good idea. I think it's a very good idea to start developing an inventory of African resources.
Yeah.
I think it's going to be quite educational for our politicians. And I think we must certainly get them to understand and accept what has been provided on the African continent. The important thing, one thing about Instruct is that the Instruct Council has not accepted proposals for membership outside of Europe at the present time. So they were very enthusiastic about Australian membership and about Chilean membership at one stage, but these were rejected by the council, and it's not being driven by the people that are actually on the ground in Instruct. So we've been actually referred to various other, well, INEXT in particular, as a more open source of assistance within the European community. The one thing about INEXT is that its funding is terminating quite soon. So if people want to use the resources in Europe, they should get on the bandwagon quite quickly. I do think that we should carry on putting pressure on Instruct to open their doors. And I also think that we should start, I saw from the Sylvia's talk that there were a lot of people involved with ICGEB, and a lot of countries in Africa that are involved in ICGEB. And I think perhaps that might be an organization that that could coordinate quite a lot of African effort.
Yeah, I was wondering what this, on the slide it said Ghana pending.
Well, Ghana pending, but I mean, do you see that even Zimbabwe has joined? I mean, it'd be interesting that's extremely interesting, you know,
Right.
the penetration of ICGEB into the African continent. And that might be an excellent vehicle to build on.
Yeah.
And I mean, it is a United Nations organization.
Yeah, sure.
So I think it's fundamentally good. I think that I don't know whether they, well, maybe they can be sensitized to a role in which they need to proselytize, in particular, biophysics in Africa. And I don't think that's so far from their mandate.
Right. Well, there were a lot of great talks in this session. So, it's not like Africa starting from dead zero. And, politicians.
The difficulty has been that most of the talks by African speakers have been actually done abroad.
That's right.
Yeah.
There's huge talent on the continent, but the realization of this talent happens abroad.
Yeah, that might not necessarily move politicians. They may say that, well, Africa can afford these things, and so, and it wasn't that Getty's sort of position in terms of people providing jobs for people, graduates of the AKC?
Well, I think that the success of science in Brazil has been promoted by government agencies sponsoring their young people to study abroad, but then ensuring that they return to Brazil. So Brazil has now become a completely integral part of the world science effort because of the contacts made by their young people abroad and their return to their home country.
How did they ensure people came home? I mean, they're not going to send goon squads, so they still have to.
They offered them jobs.
Yeah. So that's the thing. Yeah.
And I mean, even though they were in I mean, Brazil was almost bankrupt a couple of years.
Sure. And they ain't doing too great today.
But they've still built the synchrotron.
Yeah.
So they've decided that the science business is central to their further development, in spite of the difficulties associated with their economy.
Yeah. All right. Well, I got to run.
Okay. Well,
let's talk to you guys tomorrow morning.
Okay. Thanks, Elvis.
All right. Cheers.
Yeah, thank you.
Thanks, Elvis. We'll talk to you later.
All right.